committee led by Professor Frimpong Boateng at this time. And then the National Vaccine Institute, we had Dr. Sochi here today, came out. And then based on this, we were looking, okay, how can it be supported? And if you want to do local vaccine production, it is an ecosystem strengthening approach. And it is not only, it is one, you need to have skilled workers, you need to have definitely a good governance structure, you need to have a good regulator, like we have here in Ghana. FDA, I think, is the best one in Africa, I would say. You need a strong private sector, but you also need a strong R&D community. So therefore, this is our motivation, because it will never work to support Ghana to produce their own vaccines. And also, of course, we also look at the win-win situation, because we also look at the interest of, in our case, in Germany, of Europe, and through partnership, how can this be done that vaccines will be produced, and therefore is crucial to work together with research institutions. And if you talk about research institutions, our interest is not to look at research institutions as silos, not only looking at Noguchi or VACPIP or KCCR or UHAS, to find a way how the collaboration between the different research institutions can be strengthened. And I think this is something we try to collaborate. And one last sentence, we are not here to impose at all. We are here to work together and strengthen the systems.
Thank you very much for highlighting the importance of collaboration with researchers to bring advanced research and biomedical innovations. And so that brings me to the value researchers bring to the commercial space. Mr. Atule, please, what is the biggest value you think that researchers bring to the commercial space?
Thank you so much for the question. Over the years, business has actually thrived through the data that comes from the research space. Without research, I wouldn't know what the manufacturers will be doing. And I think that researchers play a very important role in using the tools and the solutions that the manufacturers produce. They actually help to validate these tools that the manufacturers make. Every tool is unique to a particular geographical area. So for instance, we have an equipment that is made for Africa based on its sensitivity and parameters, based on conditions and diseases that are prevalent in the particular geographical area. And so how do we get this data? It comes from research. So I think research has really played a very crucial role to improve the business industry of the biomedical sector.
Thank you very much. I'm sure most of the researchers seated here are quite happy to hear that their research data helps in informing policy and also production. So that's quite important to hear. And that brings me to the next. Most of us think when people sponsor scientific meetings, it's just about visibility. So Mr. Adogla, you are from a financial institution, so maybe you help clarify this. How does your company balance its commercial objectives with broader social impact?
Right, thanks for the question. I think for Stanbic Bank, one of our core focus areas is the people we serve, our clients, and the population generally. And so when we say, Ghana is our home, we drive her growth, who are you growing for? You're growing for the people, right? They are the center of it. And what makes people tick? It's, healthcare, social amenities, the things that they require, very basic things. And how do you get that done? You do that through research, innovation, and what have you. And so in our corporate social investment policy, two of our core focus areas are in education and healthcare. And what you do, what Noguchi does, falls directly within these two. I mean, you're an educational institution in research, but you're also tilted towards healthcare. And we believe that through the research that you do, through the work that you do, you bring some level of comfort to people, right? And so there's a very important part of the work that it's altruistic, making sure that our clients, through the work and the support that we give them, are able to impact our society the way we should as a business. Because if it's just about, you know, being out there making the money, who are the people bringing in the money if everybody's dying, really? You know, so at the heart of it is making sure that our people feel supported, have the right opportunities, you know, are given all the leverage they need to be the best that they can be. Because as a bank, we pride ourselves in the quality of our customers, right? Of our people. And we expect that they should be in a position where they are able to support themselves, be financially independent, and do great things. And that is one of the reasons why we support the events such as that. And we've done this over the years. But don't get it twisted. We also need you to be part of the journey that we're on, right? Growing Ghana's progress, if you like, pushing Ghana's development and making sure that Ghana is where it's supposed to be. And as a Pan-African bank, we are first and foremost who we are as a people, driving our own growth, being independent-minded, and supporting our financial growth together, which is one of the main reasons why we're here. Thank you.
Thank you. It's so comforting to know that you are not just funding scientific research for the sake of visibility, but also you care about how people feel and also improving the lifestyle of people. So that's really comforting. And as we've all already heard, these partnerships are really important to advance research, but there are also some challenges. So I would ask Dr. Akpalio, I would like to hear from you. What are some of the challenges companies face in sponsoring biomedical research in Ghana?
Thank you for that. I think that especially if you're a private sector biomedical research company, your challenges are actually a bit more as well in some instances, but there are opportunities unique as well. Some of them, obviously, everyone's talking about the money bit of it. There is that, especially if you're nascent in the space where everyone's going to ask institutionally, what is your track record? Who's going to take that first leap of faith with you from a finance or investment perspective? But fortunately, GIZ is sitting next to me, but I can say that we've been fortunate enough to have opportunities with the likes of GIZ through developmental funding. We actually were successful twice in the same calendar year last year through a developmental funding opportunity. And what that meant was that unlike your classical academic research grant options, these were actually target specific from a developmental perspective. Okay, we're going to give you, let's say, 100 CDs, and what do you expect to be able to do with that 100 CDs? As opposed to solve a research question and then publish and go from there. So on the private sector side of things, these are the ways we look at it, because the goal there is sustainability. You can't always cap in hand go asking for funding. And as a private entity, you want to quickly get to your independence as possible. So for us, funding opportunities have to be similar to what we've just gotten, where it's target specific. And that's how we have to think about it. But then there's also the other part. If you're a cutting-edge entity doing precision medicine, we say this and people ask the question. I've had people, former colleagues of mine, even in the industry in the US, ask, Derek, are you sure you're going to be able to do this? Precision medicine in Ghana, you must be mad. But we're doing it. And the you must be mad aspect of it is something that I would have also said 6 years ago, when I was in my role in J&J, running clinical trials around the globe, because there are standards that need to be maintained, and you need that validation. It's a trust space, and you have to earn that stripe every day. What you did yesterday matters not. It's what you're doing today. So for us, again, it's that evidence of stripes. How do you demonstrate that? It was why I started off with the introduction of the company as saying that we are the only entity in the region that's dual ISO accredited by the American Association of Laboratory Accreditations. And I was saying that just to establish the fact that yes, we can attain those standards right here at the first go, but importantly that what we're doing has that sustainability part, the public health aspect of it. But then once you have that, there's also the question of, do you have the skill set to be able to do that? Because again, right now in the Revna lab at this very moment, we're running NGS tests to inform treatment for pediatrics in real life today, right now. Do we have forget the equipment, do you have the skill set to be able to, one, generate quality data, two, notice that the data you're going to get will be first bioinformatic data. It's not clinical information, especially if you're using it in real time to help clinical decision making. Do you have the skill set or the tools to be able to do that conversion in real time where bioinformatic insights are being converted into clinical genomic insights or clinical insights to help with that? Yes, so that's another challenge. And of course, as you're doing all of this, we have a very progressive regulatory body in the country, but they also have to keep up, because every time you bring in a new technology, it's a new challenge, it's a new experience in some instances if the institution hasn't had the chance or the opportunity yet to evaluate that platform. So you're constantly, sort of having this tag in a way where as you're trying to advance, you also have to essentially have these entities and allied bodies go with you. And then there's the obvious one that's kind of mentioned earlier on in the day as well, where even importation of stuff is a problem, because everything we do from a precision medicine part of it is imported. Now let me just put it into context quickly and then hand the mic over. Revna Biosciences is a Ghanaian entity with US locations as well. If we order anything, call it a sequencer, for a US location and the Ghana location, by the time the equipment gets to the Ghana office, it's about 30–40% more expensive than the one in the US because of our import tax situations. Now, what do I do with that? As a scientist, there's the altruistic part of me, but I'm also a business. I have payroll to take care of, 50 employees we have right now to take care of. When you have a situation like this, what happens? You end up having to transfer these costs to the end user when, and these are some of the challenges there. And I can go on, but at least you get an idea of some of the challenges we have to go sort of deal with. So we're a bit selective about the research we do, because oftentimes the targets have to be ones that bring return quickly. We have to be solutions oriented as opposed to sometimes knowledge based. What do I mean by that? We did a collaboration with QIAGEN, for instance, on molecular assays for malaria, PCR-based assays. This was one of those that was timely, opportunistic at the time, but also with the mindset of the commercial value beyond the impact as well. So there's the impact, but as a business, I also have to think of how do we translate that into a commercial way so that we can be sustainable, and as we're sustainable, solve other problems as well.
Thank you very much. I'm sure if I ask the room who can relate to these challenges that Dr. Kwali is talking about, all hands will go up. We can all relate to these challenges, and that's why this conversation is really important today. So I'll go to the last question, Mr. Grant. Caramo has been instrumental in supporting global health and development research through strategic partnerships. From your experience, what are the key ingredients that make research successful and sustainable, particularly in low and middle income countries?
I think the primary thing is the scientific leaders. I think scientific leaders who are well connected, so partly from where they've actually done their training, where they're here, or how they've been connected internationally. So the scientific leaders, how they can lead by example, how they can mentor people, how they can change the environment and connect with their own government so that they can make things happen which, some politicians had not realized was a thing. So I think that's absolutely key, and it isn't really a low middle income country. I think that applies to any scientific research. I think the difference here is that you have to do more planning. When you're in a country where the products are readily available, as was said before, they're readily available and they can come in. It's not always in a few days. There are manufacturers who take weeks, sometimes months to produce, to turn around either do their manufacturing or bring the things into the right location. So it's not all completely smooth. It's not chalk and cheese, but the difference is you have to plan, because you're bringing in you can't be changing things on the fly and expecting to be able to then carry on with that work, because there is a longer process. The issue about importation, so Noguchi is a couple of years ago, 3 years ago nearly now, the government took away all the exemptions from all the institutions here. And Noguchi, struggled very hard and won a full exemption. Most of the other institutions have partial exemptions, so there's a cost even on the research. So even though the minister will say that they're supporting, they're then using it as a means of obtaining tax. And that's even true for Noguchi. So even though you get a full exemption, there's still a number of taxes. I think Ghana is probably at the top of the league in terms of the number of taxes that they apply to import. It virtually runs, with single lines, it virtually runs onto a second A4 page of different taxes. And at one point, I think there were 3 COVID taxes, 3 different levies, small amounts, but just one thing after another, and it all adds up. So that is a cost element, and the problem is, even when Noguchi has an exemption, they don't sign it off. So your time period just extends and extends and extends. And that's a problem for a supplier, because you buy products and you have to document a shipment for them to start the process, but products have expiry dates, and what do you do, because you documented it with lot numbers? So that's a complication. So that's a complication you have here, and I think that's probably, these days, with the way that communication has opened things up. So at the beginning, when we were working here, Ghana was relatively isolated. You had to pick up a phone. It all costs money. Now everybody can communicate in a way, and people travel in a way that they didn't do before. So I think there's less difference. The difference is the barriers that the government puts in to bringing products in, and use import basically as a source of income, because it's a very fixed thing. There's a lot of grey market out there, and they can't access that tax. So imports, well, we know what that is. We can tax it.
Thank you so much for pointing out these points. I wish the minister was still here to hear this, especially the one about the taxes for researchers, because it's a big problem that we are facing. So I want to thank all the panelists for the first round of questions, where we explored the motivations and the value proposition, and also some of the challenges. So we'll now move to the second round, where we'll talk about expectations and real world practice in multi-sector partnerships. So I'll start again with Dr. Thiel. How does your organization ensure that sponsorship goes beyond branding and contributes to Ghana's efforts in advancing pharmaceutical manufacturing?
Okay, I think to support pharmaceutical production is already our core business, but more important is how can this be kept on the high political agenda? Because as we all know, funding is for development cooperation is going down. I don't want to mention organizations like USAID and others who are no more present here. And therefore, and even now looking at development funds, there's a competition on development funds. So now, how can this be addressed to support here in Ghana pharmaceutical and vaccine production is, I think, just jointly to be successful. This is what we have to be. We have to be successful, and if we are able to show that what has been said before, that we are not only talking and talking and talking, but then that there are really vaccines being produced here, then we have something to show. If you can show that Ghana is now a regional hub for pharmaceutical production, which has been already announced also, then we are already, then we are there where we want to be. But I also have to really commend the Ghanaian government for its role, because just recently at the National Investment Forum, the president committed $50 million US for vaccine production. And this is a lot of money and is a lot of commitment. And this also helps again now, you know, if we speak now with the European Union, if we speak with the German ministry, they have much more confidence in invest more in this type of programs. And this is our role as a technical partner to make sure together with the Ghanaian partner, we make sure that the program is successful. And then I think this is the way we have to go for. And maybe one more comment is also, it is the importance of partnerships and public-private partnerships. And we should not only look at health as a social team. We have to look at also at the economic team. And this, I think, we are here sitting so many economic guys left and right. And if we are able to marry social values, social outcomes with economic issues, I think then we are on the right way. Thank you.
Thank you very much for emphasizing that GIZ is really interested to see vaccines being produced in Ghana, and you are not just funding for the branding part of it. Thank you very much. And now I'd like to hear from Mr. Tuli. What do you see as the main barriers preventing stronger partnerships between academic researchers and private companies?
Thank you for the question. Yeah. So we all know that research is time-bound, and we don't make them. The manufacturers themselves also do not make some of these things down for them to ship to you immediately they receive your payment. They need time to prepare the items. So you look at the time from where we as a private vendor makes payment to a manufacturer, and the time they receive the payment, the time they actually prepare the item for export. There are a lot of documentation that requires to be prepared for export. All these documentations are set. The shipment is dispatched. It gets to Ghana. It takes a long time for you to get it out of customs. All this takes time. From my own opinion, I think that if private companies like vendors like LabMat is being involved during the planning stage of the project, it will really help fast track the delivery and to provide the logistics that is required for the research at the end of the day. Every research is time bound. You need the items to work with. How do you get them? A researcher is working. They have run out of cell culture reagents. How do they get them? I mean, if we work closely with the researchers at the end of the day, we'll be able to understand, what are you working on? I know that Professor A is working on a project that requires, let's say, fetal bovine serum or RPMI or any of the medium. These are items that have very limited shelf life at the end of the day. If we keep them in stock and we are not sure that Professor A is coming for it, at the end of the day, I mean, it looks like juice, but we can't drink it. Yes. So we need to work closely together at the end of the day. If I know that in Q4, Prof would need these items, I add you to my forecast at the end of the day. When I'm bringing cell culture products, I add your stuff to it. When your PO comes, we just deliver the items to you. There are instances where a researcher runs out of cell culture reagents and they call, Richard, I need RPMI. So if we don't have, what happens? You lose your entire cell lines. And that's a lot of work that's have gone in there at the end of the day. So I think that if we're able to leverage on the partnership between the research and the private sector, at the end of the day, we would all be able to come together to see how can the private institution like LabMat help your project to thrive at the end of the day. Today we are here at the ARM. We are one of the sponsors of the ARM. Why are we doing this? We are doing this to help you achieve your project objectives at the end of the day. When you achieve your project objectives, I mean, this is what keeps us alive. But if you don't achieve your project objectives, we are off. Who are we supplying to? No one. So it's a win-win situation at the end of the day. So for me, I think we should leverage more on communication, timely engagement, and we should look at involving the private sector in our project planning at the end of the day. Thank you.
Thank you very much. I think I'm guilty of blaming suppliers for bringing my stuff late, and most of us here have done that at one point in time. So thank you very much for raising this point. And now we'll move to the next question about expectations. And I would like to ask Mr. Adogla, since you are from the financial institution, what are your expectations when you sponsor biomedical research meetings?
Thank you very much. I think that at the heart of every sponsorship is impact, right? We hold people's monies in trust, and they believe that those monies are being used for the right things and for impactful things. Because when we go back to them and say, look, in 2024, in 2025, this is what we did with the trust that you had in us, you are likely to get a lot more coming in. So for us, it's really about impact. Recently, we were in Ho to support the Ho teaching hospital. Apparently, they had a department that was, if you like, promoting medical tourism. And it was a first of its kind of a sort, right? So we went in there, Stambik went in there, looked at, the support that we could give and delivered. Now, what they have done with their medical support for the community is to add a layer on top of that, which is the tourism aspect, that people can come and have medical, services and have everything else on top of that, like an icing on a cake, right? For us, that is impact. You go beyond what you set to do and ensure that what you have actually told your people that you're going to do, you deliver on them. And for Stambik, that is one of the things that we drum home, even amongst our staff, that when you are dealing with your customers, you know that you have told them that you have to deliver something, right, of a certain quality at a certain time. You need to follow through on that, because immediately you miss those targets, then obviously the next time I'm coming to you, I'll think twice, right? It's like somebody who goes to the restaurant to get fufu and is served with abetia, right? They come from the same source, don't they? But they are different. They look different as well. So at the heart of it, give us impact. If we support Noguchi and you consistently deliver impact, you have. Because we believe that what Noguchi has done over the years, driving, biomedical research, if we had more funding coming in, they could do some more. And obviously I'm not a biomedical scientist, but prior to coming here, I just wanted to understand the level of funding that is available globally, right? And you would notice that between 2020, 2024 or thereabout, every year globally, the funding that has gone into biomedical science research is around, north of $70 billion. And then you dial back and look at how much of that goes into Africa, and you see that just about under 2% comes into Africa. And then if you narrow it into Ghana, then you know we're in trouble, right? But we need to understand that research offers impact. More so, medical research offers impact to our population. And we've seen all the conversations that have taken place today. Professor Genome at least gave us some more. I'm calling her Professor Genome. I'm in trouble, but I'll take it. But essentially, at the heart of it is you want the best of your brains to do the research, but they should be able to do that with the necessary funding and the backing to deliver quality, so that can morph into something valuable. Over and above biomedical research, we, as a bank, we have customers in food and nutrition science, you know, processing and all that. And all these people engage in some level of research to be able to, you know, deliver their products to the market. The best ways to actually get there is to have the right funding, the right financial advisory services, and for us, that is how we balance it. It's, yes, it's a business relationship, but it's also a partnership of a sort, that we're able to offer you the best advice, because in the end, if you thrive, the bank thrives, but ultimately the nation thrives and the people around you thrive. And for us, that is critical. That is really critical.
Thank you very much. I come from Ho, and I've seen that news article about Stanbic Bank donating to Ho, so I think I'm really happy to know that you are really about the impact and not just the branding. Thank you. And that brings another fundamental problem. Many of us seated here will attest to the fact that we wish to see our research results being turned into products, but most of the time there is a barrier. And so, Dr. Akpali, I would like to hear from you. Sorry. In your opinion, what are the key ingredients needed to successfully translate research findings into market-ready biomedical products?
I think for us, or for me, the answer is very simple. If the focus is solutions-based, what question are you trying to answer? What problem are you trying to solve? If that question is asked with the scientific rigor that you need and backed with the right pace, then it's likely to be of good commercial value. And I use that word here loosely, where commercial value is you're solving that problem, you're providing the impact, and from an IP perspective, intellectual property perspective, that becomes a marketable tool. So for us on the Revna Biosciences side, that is what actually drives us. What problem are we trying to solve? Why hasn't it been solved? And why now? If you go through that process, you will have solutions that become, one, impactful, because we all use the word impact, but what is really impact, right? There's a utility to it, there's a timeliness to it, and that relevance as well. And if you can check those boxes, oftentimes I say to the team, don't think of research or the money or the investment that goes into research as sunk cost. It isn't. It's an investment into that's something you can leverage, and basically we see this as we're beginning to build disease models, where we funded research activities or collaborated with partners to solve a problem, and now you're getting these phone calls saying, hey, Revna, this issue is becoming a bit more urgent. How quickly can we get this out of the research phase to that? So for us, it's about doing those things that allow you to translate very quickly from the bench side, so to speak, to the market. If you're solving real problems, oftentimes the commercial aspect of it will take care of itself. And this was a lesson that I learned in the industry, where it was basically this: where clinical goes, commercial goes. Essentially, if the clinicians, the researchers, the scientists solve the right questions, the commercial people will find a way to commercialize it. And that's my response to that.
Yeah, thank you very much for outlining those critical elements. And Mr. Grant, given Karamo's long-standing commitment, how are you positioning yourself to support the next wave of innovation, especially in tech and data-driven research in Africa? Especially in tech, technology and data-driven research.
So it's a complicated field. I mean, there are so many vendors. What we do is there's a number of us who are scientists on the commercial side dealing with the quotations. So what we're doing is partly in combination with the customers, where the customers are coming to us with a new innovation that they've read about, or there's been a paper, or they've been at a conference and there's been a discussion. so what we're doing is using that information alongside those conversations that we have with our vendors, and then telling us about their new applications, their new instruments. And a good example was Nanopore. So Nanopore, we came to Nanopore, both we came to them and they came to us, partly through some of our existing customers who were exploring that technology in sort of 2017, not long after they actually commercialized their first, MinION. And there were lots of issues going on, and a number of customers had suggested that they approach us. We then had a long conversation, and because they're quite legalistic and looking at IP, it took about 1.5 years to form an agreement, and that was obviously quite crucial, that relationship. We started saying we will deal with that technology into a number of countries, which was going to include Ghana. That was the end of 2019, and then of course the pandemic arrived, and all bets were off, and it just went crazy. And at the same time, the problem was that not so much the Nanopore supply. They were ready for that. It was all the other consumables and products that go alongside the technology. That was the challenge during the pandemic. So it's this combination of talking to existing vendors about what they're doing, so understanding where we can actually potentially suggest, a product to a customer, or it's customers coming to us and us exploring that technology and connecting with a new vendor. So we're not tied. We're adding vendors all the time, and that's really just part of the process. And who knows where it goes.
Thank you very much. Please, let's thank our panelists for this second round of discussion. Thank you.
Thank you. We are almost at the end of our session, but I think I'll invite our MC, Christopher Abana, to give a brief announcement.
Hello. Yes. Thank you so much for staying with us all this while. We are grateful. We understand some people have issues with posters. So if you have an issue mounting your poster, kindly visit the registration desk downstairs to mount your poster just before we go for lunch. Please don't forget to do that. Right after here, we'll be having lunch. There will be breakout sessions at 3 different areas. We have the West Africa Genomic Center here just behind us. Then we have the seminar room here, and then a combined seminar room as well. Then there are QR codes that have been posted around the wall in the seminar room here. Kindly scan for the book of abstract as well. Then in order not to miss your emails and send that text message or WhatsApp, kindly connect to our Wi-Fi at ARM 2025. You can see the passwords just around the wall. Thank you so much.
Thank you. So I will now ask the last question in this session. And we just want to know, how do our panelists today see the role of sponsors in biomedical research evolving in the next couple of years, in 2 minutes? So please.
Yes, I'm lucky. Yes. I get to pick. and the answers run out. Anyway, so based on how things have gone, especially even looking at the geopolitics around, we mentioned, some of these legacy funding entities that have literally disappeared in 2025. There are alternate models that are springing up that we all need to look at. What we're seeing is a shift, one, towards the more milestone-based, results-oriented kind of funding model where, okay, this is the total amount, these are the timelines, but we want that developmental impact measurable in phases. So as you earn or as you make those, you get more, you keep going till you get to the end. So there is that. But what you also find is that there's the need, or there's a drive towards a bit more of sustainability. So you're finding that funders are no longer being philanthropic in that sense. They're participating a bit more in co-development of the projects. They're also vested in measurables, the sustainability of it as well. So basically, how scalable is what you're doing in its instance, and then also that sustainability part of it, which is not just academic, what are you solving and how is that then used to do something else? For instance, the GIZ is pushing very hard on the vaccine development role. What would a Revna or a Noguchi do in that space? There's a whole lot of life sciences that goes in there. What projects are we picking? What questions are we trying to answer that others in that space can then pick and then run with? So that there is a bit more of intentionality in terms of how they're funding and a bit more obligation on awardees to actually put those funds to good use as well. So I think I'll stop there and leave it.
Okay, I will actually not go so much in answering your question, but I will just mention one topic and do one announcement, if you don't mind. But it's an example also where Noguchi is involved. I think what is very important if you look at research and also the future of research, especially now and I think it's not only in the area of vaccine and pharma production, is the importance how research institutions are working together with the private sector. And and I just give you an example from our PharmaVax program. We did a so-called call for proposal where research institutions were invited to bring their proposals, but there was a condition. There has to be a key partner from the private sector. If not, your whole proposal will be definitely not even considered. So after the call for proposal, there were 43 proposals, and out of them were 31, if I'm not mistaken, 31 which were eligible. Very good quality, and now it's coming as an announcement. So you might ask, was Noguchi successful, yes or no? I don't know how the answer will be, but of course they were successful. So, and this is, for instance, it is an initiative where Noguchi, and I can even mention it here, is partnering together with Atlantic Life Science, which is one of the vaccine producing companies. I think they brought a proposal over 500,000 euro in the area of tetanus, diphtheria, vaccine stability testing. So now, of course, it has to go through a commercial eligibility test, and if it's positive, then this project can move on, and this could contribute directly to what is at the end. The aim is that a local vaccine can be produced here in Ghana, which would be the first one. So this is one example, and congratulations to Noguchi, by the way. Thank you.
Okay, so thank you so much. Thank you. Yes, so thank you for the question. I want to speak briefly about the involvement of the private sector in supporting research in Ghana. And we all know that beginning last year, the funding space has become a bit shady, and that makes it very difficult for researchers to be able to come up with enough resources to be able to purchase all the logistics and to conduct the activities that they are supposed to conduct. Now the big question is, what will be the role of the private sector, the private partners that are working with the research institutions? What value can they add to these institutions to enable them or to empower them to be able to achieve their objectives? And this is why we are all gathered here at the ARM 2025. And this is why we have taken the initiative to support Noguchi in the ARM 2025. We know that Noguchi has several projects, and in each of the projects you have your funding under it, but each funding has an objective. I don't think there is any of the objectives that states that you should use part of that funds for ARM 2025. So we believe that, yes, you empower us, we also have to help you achieve your project objectives in the little way that we can at the end of the day. And then lastly also, I want to talk briefly about student projects and the the impact that LabMat is also creating on students' projects. I think we talk about scientists, we talk about innovations, but we forget that all these happens when these young scientists are being groomed to a certain stage. What are we doing for these young scientists at the end of the day? To be honest with you, when you enter the labs of Noguchi, you see innovative instruments, high-end equipment. Trust me, there are a lot of institutions in the country that you enter, not even a centrifuge. A centrifuge, you will not even find a centrifuge in the lab. But they are doing biomedical laboratory science. You go and sit on the lab bench in the lab, go through a session, and come out. No practicals, nothing. So LabMats, it's actually working closely with students to be able to assist them with their projects at the end of the day. Sometimes students have very innovative topics at the end of the day, but how do you come up with logistics to be able to achieve those objectives? And sometimes they don't even know what it takes to be able to run or to be able to complete that particular project at the end of the day. They select the topic, the tools that is required, their institution doesn't have it, the reagents that is required, they don't have the resources to be able to purchase those reagents. How do we come in to assist? We have a team in-house that is able to work closely with students to understand what is your project objectives, what logistics do you need, and we check internally to see which of the many vendors that we partner with would be able to provide that particular reagent or product at a reasonable cost for the student to be able to afford at the end of the day. We think that this is the little way that we can also impact science generally. And in the end, we all produce the future generation of the scientists that we are actually aiming for. Thank you.
Thank you very much. If this was a classroom setting, I would have said, they've said everything, so that's it for me. How many of us in here have a relationship with Stanbic Bank? Okay. Yeah, not that bad. At least there are people here, but there's room for improvement. Before I end, I'll invite you over. So to your question, I think that going forward, sponsors are going to be looking at two critical things. It will be based on values-driven partnership. So we will not step out there to sponsor anything or everything just because it's for the public good. But is it values driven? Are you doing what you're doing based on a certain level of belief, passion, and drive? If that connects, those values connect with ours, I believe that that is how the world is navigating towards, because we want to drive things that meet our expectations. Recently, we did some work around our sustainability team or unit, and the focus was, in spite of the fact that we've done a lot of work over the years, we want to pivot around the things that matter to us. And the things that matter to us are things that help drive Ghana's growth, pure and simple. So if you're doing whatever research, whatever ideas you're running with, it must be around the values that we share in common, right? The other thing for me is about big data. Now, research cannot sit away from big data because they speak to each other. And we will drive sponsorship based on what the data says about what it is that you want to drive. Is what you want to implement, feasible in the long term? What do we hope to see, or what kind of value do you expect to see? There would be a lot of computation. We just want to make sure, and I'm speaking as a potential sponsor in the future. I want to see real value, looking at the values that you present as an entity, and then also around the data that supports the work that you do. And then I believe that we would both see value together. At the heart of it all, I want to invite all of you, those who are not yet, having a relationship with Stanbic Bank, please. From here, we have a branch on campus. Try and visit them, and I believe you will smile. Thank you very much.
I agree. Much of it has been said. I mean, the environment has changed. So, I mean, I remember 10 years ago, nobody asked for sponsorship. You know, the Gates Foundation, the Wellcome Trust, they sponsored it, you know. And it was only later on that they suddenly realized that there was an opportunity, or that part of the funding, and was, how are we going to do it? And so I'm given that the funding environment has changed so radically this year, I was at a meeting in June with Gates and Wellcome Trust. They cannot make up that gap. You know, that they're being asked to do that, but they're going to reprioritize because they can't fill that space. That face is too big. You know, and it's obviously affected Gavi. It's affected WHO. So it's really affected the space. So I think it just means that yes, you need to talk to your sponsors. You need to talk to commercial companies, but even in your research sort of ideas, you need to be more spread out. I mean, those, probably, Noguchi is probably, lucky in the sense that it's not so massively embedded in the United States, but those people that were very embedded in that, this is a year of crisis, some people are lucky if they've still got their jobs. You're in a, not in such a difficult position, but it is a difficult time, and commercial sponsors obviously a way to allow you to be able to run events as good as this, with that support. So, but in terms of your research side, you need to be spread out, in terms of your funders, because who knows what thing will happen next.
Thank you very much to our panelists. This has been very insightful. I'm sure all of us here have heard a lot of information, but I think what we can take home today is the idea of collaboration. We've heard a lot of things about collaboration, involving partners more, like from the inception to the very end. We've learned a lot about being intentional about the type of science that you are doing, and I hope that as all of us are sitting here today, we've taken something from this panel which will help us in our future endeavors. Thank you very much again to our panelists, and have a good rest of your day.
All right, thank you so much, Charlene and Edith. We are so grateful for the opportunity. And to all our panelists, thank you once again. Thank you so much. Please, we are not done. We are not done yet. Kindly take a seat. Yes, right after here, there's lunch, and the lunch is combined with poster session as well. You heard from the organizing committee that there are going to be awards. So if you have a poster, kindly make sure you grab your food quickly, and kindly make sure you stand by your poster, because someone may come and talk to you. And lunch is only 40 minutes. So please kindly grab your lunch quickly and get to the poster session as well. And like I said, right after lunch, there are going to be breakout sessions. We have the main conference room here, the combined seminar room, and just behind us at WACMEC, the ushers are going to direct us, those going to WACMEC, those going to the conference room, or even the combined seminar room. And if you are chairing a session, please check to see where you belong. And also what interests you, the kind of research that interests you, kindly make sure that you go to the right room for that particular session. Thank you so much, and see you at lunchtime. Thank you.
Team, what do you think about our upcoming debate?
All right, so we are excited about this debate. This forms part of the 25th anniversary celebration of the College of Health Sciences, and we are gearing up for this debate.
Team Hollywood is truly excited. We know we are going to win. We are surely going to prove that digital healthcare will widen the equity gap.
And even as we win this debate, it's not just a victory for us, but a wake-up call to Africa to start building real foundations for our healthcare.
Yes, it's going to be exciting, insightful, and I'm excited because the points we are going to turn out will shape how healthcare is delivered on our continent for many years to come.
Digital health will certainly widen the healthcare equity gap in Africa, and there are lots of insights to be revealed from this debate.
The debate comes off on Thursday, the 27th of November at 2 p.m. at the Great Hall of the University of Ghana campus, Legon. See you there.
Hi everyone. As part of the University of Ghana's College of Health Sciences 25th anniversary celebrations, there's going to be a grand debate on the topic, digital health will widen rather than bridge Africa's healthcare equity gap. And we are against the motion. As lead, my name is Dr. Lily Pemka. I'm the Deputy Director at the West African Genetic Medicine Center. I'm a molecular geneticist.
And my name is Dr. Justice Kumi, a supporting speaker from the Noguchi Memorial Institute for Medical Research, and I'm a toxicologist.
And I am Dr. Nana Amamrukujima from the School of Pharmacy, and I'm a natural products researcher.
I am Dr. Constance Agbemolutumafu from the Noguchi Memorial Institute for Medical Research. I'm a molecular biologist, and I'm a supporting speaker.
And we are
Team Legon. This debate is happening at the Great Hall on the 27th of November 2025 at 2 p.m.
Come, let's have fun. Come and let's learn digital health from Ghana and Africa and beyond to all parts of the world. And we know that we are winning. Catch you there.
Come and be wild.
We are winning.
We are winning. We are winning.
I welcome you to the 10th annual research meeting of the Noguchi Memorial Institute for Medical Research, College of Health Sciences, University of Ghana. This year we have a lot for you, and the theme for our annual research meeting is Advancing Healthcare Through Impactful Research and Innovations. This ties in very well with what we are doing. Noguchi, we support health delivery not only in Ghana, but in the sub-region. We employ you to join us. We have innovative speakers who are going to speak about their innovative research, antimicrobial resistance. Why is that you go to hospital and they give you all the antibiotics, but you are not responding? Please come, get understanding. How do we prevent the spread of antimicrobial resistance? Please, there are so much information for you. Malaria, is it coming down? Is it going up? We are there. Come. Innovative solutions, drug discovery, diagnostics. So I'm employing you. Come, join us. 25th, 26th. It is not going to be science alone. We are going to have fun. We are inviting all school children. We are going to open the doors of Noguchi. Come and look at all the machines heard about. JHS, whether you are in primary school, secondary, tertiary, the Noguchi doors will be open to you. Come and look at innovative, the infrastructure that you've been dreaming about. Come and listen to our impactful research that is making waves across Ghana and the sub-region. For Noguchi, there's nothing but quality research.
Hello, my name is Professor Chijo Kamitu Souza. I am from the Noguchi Memorial Institute for Medical Research. I'm the co-chair with Professor Kodjoa Sabanusi planning this year's annual research meeting of the Institute. This is our 10th annual research meeting, which also coincides with the 25th anniversary of the College of Health Sciences. So this research meeting is being organized together with all other constituents of the College of Health Sciences, bringing together all the scientists to showcase the work that we have been doing.
The theme for this year's annual research meeting is advancing healthcare through impactful research and innovation. And we are having this program from the 25th to the 27th of November 2025. We are having on the first day a keynote to be delivered by one of our own, Professor Beverly Ejim, who will be speaking on her work in AMR. We will have, as part of that day, also a panel discussion where we would have some of our sponsors who would speak about the impact of private-public partnerships in promoting research and innovation. Our parallel sessions happening in the afternoon to bring the first day to an end. On the second day, we begin the day with a symposium, a special symposium by NMIMR, that is a JICA-funded program that is based at Noguchi. They were having a one-hour symposium to talk about some of the research that they have conducted over the past years. And then we would have our poster sessions beginning with a table session where selected posters will have 2 minutes to pitch their ideas and invite others to join them for the afternoon session. Then we continue with our parallel sessions in the afternoon for the second day. On the third day, which is one of the unique things that we are doing this year, we are having a joint celebration of our annual research meeting with the College of Health Sciences 25th anniversary, and we are having a joint activity which will be hosted at the Great Hall. And in the morning, we will have the closing session for the climax of activities for the College of Health Sciences, and then we'll continue with activities relating to our annual research meeting. First, with a keynote session from Professor Abdullah Jimbe, who is the Noguchi Africa Prize winner for 2025, a researcher with the University of Akko in Mali. He will be here to share with us on his work in malaria genomics and how we can transmit findings from the bench to the bedside. We will end the day with another unique thing we are doing this year, which is our annual debate.
You do not want to miss the debates. It is Legon versus Colegon. And we would want to see who will win the debate. It's about digital technologies and health in the current age. And whether digital technologies would bridge the gap or extend the gap in healthcare and access between the rich and the poor, the have and the have-nots. We aim to give awards to young scientists who will be presenting their posters and their abstract, so at least there will be a prize, first, second, and third prize for the abstracts and the poster presenters. The open day, we are expecting at least 1,500 students from southern Ghana. Most of them are from within Accra, but some are from the eastern region, others from the western region. And this day affords the opportunity for young scientists, the future researchers, to get to learn about what we do, see our labs, learn about our equipment, and hopefully be inspired to become scientists in the future.
Going through the 9 departments, immunology, virology, so it's nice to find out the kind of jobs you can take on by learning, and show that your learning is not really for nothing. You can interact, ask questions, and just have a good time and meet new people. So it's been a lovely experience.
This promises to be explosive, and we are inviting all of you to come and witness this as we climax the activities for our annual research meeting this year. We thank all our sponsors for their dedication and commitment to us and for opening their doors when we reach out to them for support.
Africa stands at a crossroads. Technology is transforming healthcare like never before. But will digital innovation bring healthcare closer to everyone or leave some even further behind? As part of the 25th anniversary of the College of Health Sciences and the 2025 annual research meeting of NMIMR, we are bringing you a live thought-provoking debate where two of the brightest teams face off. Experts from Team Legon versus experts from Team Kolebu.
Expect fierce arguments, fresh perspectives, and powerful insights that must reshape how we see the future of healthcare innovation in Africa. Trust me, this is one conversation you do not want to miss.
And the motion, digital health, will widen rather than bridge Africa's healthcare equity gap. And this is happening on the 27th of November 2025 at the Great Hall, University of Ghana, 1 p.m. sharp.
Be there, be inspired, because the future of African healthcare starts with the conversation we have today. See you at the Great Hall.
Dear team, what do you think about our upcoming debate?
All right, so we are excited about this debate. This forms part of the 25th anniversary celebration of the College of Health Sciences, and we are gearing up for this debate.
Team Kolebu is truly excited. We know we are going to win. We are surely going to prove that digital healthcare will widen the equity gap.
And even as we win this debate, it's not just a victory for us, but a wake-up call to Africa to start building real foundations for our healthcare.
Yes, it's going to be exciting, insightful, and I'm excited because the points we are going to churn out will shape how healthcare is delivered on our continent for many years to come.
Digital health will certainly widen the healthcare equity gap in Africa, and there are lots of insights to be revealed from this debate.
The debate comes off on Thursday, the 27th of November at 2 p.m. at the Great Hall of the University of Ghana campus, Legon. See you there.
Hi everyone. As part of the University of Ghana's College of Health Sciences 25th anniversary celebrations, there's going to be a grand debate on the topic, digital health will widen rather than bridge Africa's healthcare equity gap. And we are against the motion. As lead, my name is Dr. Lily Pemka. I'm the Deputy Director at the West African Genetic Medicine Center. I'm a molecular geneticist.
And my name is Dr. Justice Kumi, a supporting speaker from the Noguchi Memorial Institute for Medical Research, and I'm a toxicologist.
And I am Dr. Nana Amamrukujima from the School of Pharmacy, and I'm a natural products researcher.
I am Dr. Constance Agbemolutumafu from the Noguchi Memorial Institute for Medical Research. I'm a molecular biologist, and I'm a supporting speaker.
And we are
Team Legon. This debate is happening at the Great Hall on the 27th of November 2025 at 2 p.m.
Come, let's have fun. Come and let's learn digital health from Ghana to Africa and beyond to all parts of the world. And we know that we are winning. Catch you there.
Come and be wild.
We are winning. We are winning. We are winning.
I welcome you to the 10th annual research meeting of the Noguchi Memorial Institute for Medical Research, College of Health Sciences, University of Ghana. This year we have a lot for you, and the theme for our annual research meeting is Advancing Healthcare Through Impactful Research and Innovations. This ties in very well with what we are doing. Noguchi, we support health delivery not only in Ghana, but in the sub-region. We employ you to join us. We have innovative speakers who are going to speak about their innovative research, antimicrobial resistance. Why is that you go to hospital and they give you all the antibiotics, but you are not responding? Please come, get understanding. How do we prevent the spread of antimicrobial resistance? Please, there are so much information for you. Malaria, is it coming down? Is it going up? We are there. Come. Innovative solutions, drug discovery, diagnostics. So I'm employing you. Come join us. 25th, 26th. It is not going to be science alone. We are going to have fun. We are inviting all school children. We are going to open the doors of Noguchi. Come and look at all the machines you heard about. JHS, whether you are in primary school, secondary, tertiary, the Noguchi doors will be open to you. Come and look at innovative, the infrastructure that you've been dreaming about. Come and listen to our impactful research that is making waves across Ghana and the sub-region. For Noguchi, there's nothing but quality research.
Hello, my name is Professor J.J. Komi D'Souza. I am from the Noguchi Memorial Institute for Medical Research. I'm the co-chair with Professor Kodjosa Wakusi planning this year's annual research meeting of the Institute. This is our 10th annual research meeting, which also coincides with the 25th anniversary of the College of Health Sciences. So this research meeting is being organized together with all other constituents of the College of Health Sciences, bringing together all the scientists to showcase the work that we have been doing.
The theme for this year's annual research meeting is advancing healthcare through impactful research and innovation. And we are having this program from the 25th to the 27th of November 2025. We are having on the first day a keynote to be delivered by one of our own professors. As part of that, there also a panel discussion where we would have some of our sponsors who would speak about the impact of private-public partnerships in promoting research and innovation. Our parallel sessions happening in the afternoon to bring the first day to an end. On the second day, we begin the day with a symposium, a special symposium by AMEC, that is a JICA funded program that is based at Noguchi. They were having a 1-hour symposium to talk about some of the research that they have conducted over the past years. And then we would have our poster sessions beginning with a table session where selected posters will have 2 minutes to pitch their ideas and invite others to join them for the afternoon session. Then we continue with our parallel sessions in the afternoon for the second day. On the third day, which is one of the unique things that we are doing this year, we are having a joint celebration of our annual research meeting with the College of Health Sciences 25th anniversary, and we are having a joint activity which will be hosted at the Great Hall. And in the morning, we will have the closing session for the climax of activities for the College of Health Sciences, and then we continue with activities relating to our annual
education and stuff, like who important and stuff, individuals can actually input in the data, and that will help in the progression between treating your cancer if we help once it detects. But the challenging thing is about the imaging, because the model has to be great, and right at the moment, since our model is quite and dicey, 85% and 5%, we didn't try to now push it out there. So now our work is now more on optimizing this. Then once we fully optimize the biomarkers, we can actually try to put it all the other ways for next implement and work. And our challenging issue was on data set. I knew you were going to say data set is very quite there because everyone Irish in different countries is very different. So you have to be getting Ghana very different. There's some differences. So one of the things we want to focus on is we want to centralize it in Africa. So we don't want to go outside or something, because if you go outside, there'll be so many. It will be very, very trouble. Because one case also, one guy presented also on biomarkers on here, and he was having a lot of trouble explaining because he used a lot of data from different variations, which wouldn't match at all. Your accuracy score. So that's why I thought about using it at the moment because that score wouldn't be
Secretary,
very
should I stay till he's done or I should move to another person too?
Let's break that.
But let me invite you to another project that we talked about. Yes. I think a non-profit. Yes, yes. Yeah.
Okay.
I discussed with him like where we can market and we are looking at videos at the moment. Using that's why your community system. I was talking about your engagement. You have to put it with yeah, but we need to use those seeds or greater the model and use it to study the various demand and be able to predict accurately. This is a rule yeah, well, this is a wound of secondary infection. Yeah, which wound is just a wound which burns or something. So well, now you actually have to get the law immediately for that is banned and also that's yes.
Is it okay now?
And you have to get the immediate that have been seen by the water. Okay, you said that aside, but the issue is getting data. That's always a big question in actually trying to treat one. Getting the virus and use data to then that's nice. So you have to use this. So we can use this here, but we need a question. Then you can show you based on the weeks you confirm these things that you are based on PCR or like to think of that. Then you train the model to know I'm more focused on the computer. When you get a system or and to test whether it's a computer. Actually everything is on the web where you get the final goal of your research is your mobile app. You can actually do on the mobile asset back in you get and then get those that's fine. How we can push it. Yeah, but currently, as I mentioned, this is on the also have the few councils have to expand. It's going to be very because you have to again. So the new data we have to do something. It's from Paradise. I know you know Paradise. I think and also that was Paradise. The one I love. Yes.
Hello.
That's charge. The staff of those. Most intense we're not actually planning.
Can you please start making our way to our various breakout sessions? We have one here in the conference room, and that is the theme for the talks here in the conference room is tackling infectious and non-communicable diseases. There's another session going on in the combined seminar rooms. And that's on strengthening health systems for resilience and sustainability. And there's a third concurrent session, and that's on maternal and neonatal health. And this will be held at the Ragnick Conference Room. And for those of us who are interested in going Ragnick, there's a bus in front of the conference room shuttle us to Ragnick. The time starts making them to the seminary. Have some of the actions here in the main conference room.
So this is a mixture of how they how everyone.
Our presenters move to the various breakout sessions. If you're interested in tackling infections and non-communicable diseases, please move to the conference room. We are about starting. There's another session going in the combined seminary room, and that's on strengthening health systems for resilience and sustainability. And the third session on maternal and neonatal health and rhythmic conference syntax.
That's where those bacteria.
The bars in front of our conference room to shuttle people to the rhythmic conference room.
When
one make it across the street.
We are coming to this way.
From the poster assessors, can we have all presenters and all participants please move to the various breakout rooms?
Yeah, so you're welcome. You're welcome. My sister.
We'll start on time so we can finish early and schedule.
As we all know, NMIMR is a global health challenge. There's a growing global concern over pseudomonas aeruginosa as a major cause of nosocomial infection. And this challenge is heightened or worsened by the high prevalence of multi-drug resistant pseudomonas aeruginosa strains in our hospitals. If I say our hospitals, our local hospitals, our regional hospitals, hospitals. And this complicates treatment options and worsens patient outcomes in our hospitals like I mentioned. So, in view of this, we wanted to look at this resistant profile, genomic characteristics, and transmission dynamics of pseudomonas aeruginosa from clinical sources across 7 hospitals in Ghana. So, we work with Lexma Hospital, Saints, and Martin de Forest, and Patrick Hospital, University of Ghana Hospital, and Tamale Teaching Hospital. So basically, we've partnered with them. Routinely, they test these patient samples, blood, urine, wound swab, ear swabs, semen, and what have you. So whenever they recover pseudomonas, they cryopreserve, get isolated, and they are shipped to our lab.
For those of us interested in maternal and neonatal health, the talk should be held at the rhythmic conference room, and that's ready to move right in front of the conference room to Rhythmic. If you are coming to Rhythmic, there's a shuttle ready, and just about leaving.
And then we proceeded to do DNA extraction using the Cajun, following the Cajun protocol. And then we did some library preparation. We fragmented the DNA, and then we tagged with barcodes, and then they are loaded, they are put together first, they are loaded onto the Illumina mindset for sequencing. Once the sequencing is done after 78 hours, we get raw rate Pacific files, analyze these sequences using chemomatic stream of bad rates. We also use FastQC to do some quality control or do quality check then we use the Unicycle to assemble the content from the library preparation. Because here and they are fragmented. So we have to pull them together to form the whole genome of the organism so that we can better understand the features. So after assembling using Unicycle, we proceeded to use various online platforms from the Center for Genomic Epidemiology to look for resistant genes, look for virulence genes, look for multiple sequence diet, then other mobile genetic elements. So we also perform some phylogen, look at the transmission dynamics of these organisms. Now for
whole genome sequencing.
yes, whole genome sequencing. So after analyzing the phenotypic data, the antimicrobial susceptibility test results, we realized high level of resistance to ciprofloxacin, eropinem, piperacillin, tazobactam, gentamicin, and then the rest. Now, of interest here is meropenem. Meropenem is a last line treatment drug, which is quite expensive in our hospital setting. Then, a patient goes to hospital, is diagnosed with bacterial infection, they pick sample, they do all these processes, they do AST, and you realize that the organism is resistant to almost everything. Now, they pick
because you have different products. I'm using some steel. And then also,
My colleague is bringing something. Can I take that to you?
Are they polycombs or monocombs? Because monocombs just have one color. One color, and then polycombs have like multiple colors. So ground staining is actually come from the inside, whereby it binds to the nucleus and then the cytoplasm. But ground staining, I actually want to know polycombs. And then also how it's actually happening. That's it. What are you talking about? And then the demographics, I think it's not that one. So that one's fine.
So your first question, can you repeat your first question?
Yeah, so you said, so the list or this one, and then the ground yes, or you have it, look at this one like that, it shows ground staining because that must involve.
Vedic media. You've heard of Noguchi, right?
Yeah, I've heard of it.
Vedic media. So allow any fan group. It doesn't give a particular color. It doesn't work like this. This is a homogenic media. The homogenic media has substrates on them. The organism produces certain enzymes. So the enzymes react with this substrate to produce color. So the organism will go on this media with different colors. And that gives you a presumptive idea of what you are working on. But this is a basic if it's in list, they are going to be creamy or white sections. They are not going to be a particular color. Now, there are different stages that you can do. We have huge, that's potassium hydroxide, or potassium hydroxide. These things what is show you the yeast cells. You know, you could eat white blood.
So please, we have breakout sessions. We have been asked to move to the conference room. And after the breakout sessions, we can come back for the post. Time for so I go to some partnerships within the current authorities in place. So I think it's maybe a bad idea. Then you're also starting this.
Stop it. You're not that.
We are now in what about you?
Okay, thank you.
It's time for presentation.
So what I'm saying is, so this reading, you understand, they are all to go right.
It's time for presentation.
I'll show you. 1224. That is between 400 and 500. If you don't have that, and these things are not aligned, you can't tell whether D and D are.
It's time for presentation.
Okay. Thank you.
You're presenting someone. You're talking to someone.
Good afternoon once again. All welcome to another session.
Do you mind if I explain one of the
Why you say we should go to the listing, so maybe after.
Okay. Thank you.
Please, as you file into the room, kindly move forward and fill the spaces at the front rows. Please come forward. Please come forward. Please come forward. We ask you to come. Please come forward for us. Please come forward and fill the spaces at the front row, please. Okay, thank you so much, and you're all warmly welcome to this afternoon's plenary session. We have two chairs for this session. The first chair is Dr. Hiroyuki Yamamoto of the AIDS Research Center of the National Institute of Infectious Diseases in Tokyo, Japan. Please, let's welcome him.
My dear colleagues, this is Hiroyuki Yamamoto from the National Institute of Infectious Diseases, Tokyo, Japan, And we have had a 10-year exchange of academic research on our department basis. And Dr. Chris, he also has been like a previous temporary colleague in conjunction with JICA. And it's really, really an honor to join you. It's my very first time in Ghana, and I really, really respect the vibes of this rising country. Yes, and I'm really looking forward to like link with you on many portals. Thank you so much.
Thank you so much, Dr. Yamamoto. So we also have Dr. Ivy Asante of the National Influenza Center at the Virology Department of the Noguchi Memorial Institute for Medical Research as a co-chair.
Hello everyone, good afternoon and welcome to the session. And we hope to have an exciting session, please. I think you all know me. I work here at Virology Department. Thank you.
Thank you, coaches. Please take the charge. Thank you.
All right, thank you once again and welcome to this session. As the director in her speech mentioned, Noguchi's mandate is in threefold. The first one is to conduct research into infectious and non-infectious diseases. And for this session, our title is Tackling These Infectious and Non-Communicable Diseases. And we have a cocktail of beautiful topics for you to digest. So please pay attention so that we can have interesting discussions. And so to start, Doctor, do you have some more remarks? Okay. All right. So to start, there are a few things to note. First of all, we want everyone to present. So after your presentation, then we'll have a question and answer session. So for the presenters, you have 10 minutes to present. When you have 2 minutes more to go, I will wave my sheet to let you know that you have 2 minutes more to go. Please, everyone who knows me knows that for these things, I'm very good at it. So that you will stick to 10 minutes so that we are done for your presentation by 10 minutes. And then we'll have 5 to 7 minutes of discussions, please. So to not waste too much time, our first presentation is from Nathaniel Opoku. Nathaniel will present to us on Staphylococcus aureus co-colonization with other Staphylococcus species in commercially raised pigs in Accra, Ghana. Please, let's give him a round of applause. And all the best to you.
Good afternoon to you all. I want to address the chairperson and stand on all established protocols to deliver my presentation. I'm Pokubo Nathaniel, and I represent Staphylococcus aureus co-colonization with other Staphylococcus species in commercially raised pigs in Accra, Ghana. This is going to be the outline for my presentation. The increase in antimicrobial resistance of Staphylococcus species in livestock highlights the need to understand their spread and colonization in pigs. Staphylococcus species, often a Staphylococcus aureus, often colonize pigs with a methicillin-resistant Staphylococcus aureus, posing a major threat to human health. Livestock-associated Staphylococcus species is common due to their evolved antimicrobial resistant patterns. In pig farming, the use of antimicrobials as prophylaxis and for growth is increasing due to consumer demand and regulatory pressures. Our study sought to determine the prevalence of Staphylococcus species among commercially raised pigs in Accra, Ghana, also to assess the antimicrobial resistant patterns of Staphylococcus species and to determine the association between multi-drug resistance and farm practices among the pig farms. So our study employed a cross-sectional study design using Accra as its study site, sampling 274 pigs from 20 commercial pig farms. And data was collected through sample collection and sample analysis to run through our study. And the sample type we used was archived nasopharyngeal samples. And for our inclusion and exclusion criteria, we involved only healthy pigs in our study. So first, our archived nasopharyngeal samples were cultured in 5% sheep blood agar, which were further cultured on mannitol salt agar, and gram staining was performed on these isolates. To separate between our coagulase positive and coagulase negative staphylococci, we performed the coagulase testing, and for our coagulase positives, we performed cephosity sensitivity test to find the MRSA among the positive staph isolates. And for our coagulase negative staphylococci, we performed the vaccine sensitivity test to separate our Staphylococcus epidermidis from our Staphylococcus saprophyticus. And finally, we performed antimicrobial susceptibility testing on all our isolates. So looking at the characteristics of the farms we involved in our study, we had 274 pigs involved in our study, of which the majority were males, 145, and females were 129. And for the age range, it was between 3 to 5 months. So 4 months were the highest among the pigs we employed in our study. And for the breeds involved in our study, they were all exotic breeds, and there were no local breeds involved. And all the farms confirmed using concrete pens as housing systems. Also, we took some data on some farm practices, which include antimicrobial use, and all 20 pig farms confirmed using antimicrobials. And they also confirmed the presence of other farm animals on the farm. Other than that, too, they also confirmed the visit of other animals from external sources, and also they confirmed disposing their waste on the farm as well, of which 18 out of 20 farms confirmed disposing their waste on the farm, and 2 confirmed disposing it outside the farm. So we also took some data on the type of antimicrobials used, and we can observe that the highest antimicrobials used among the farms were penicillin-based antimicrobials, which 13 out of 20 farms confirmed using, followed by sulfonamide trimethoprim, 9 farms out of 20, and tetracycline, 8 out of 20, and tyrosine tartrate, 4 out of 20, and rifloxacin, 2 out of 20, on which the least used antimicrobial is gentamicin. Only 1 farm confirmed using gentamicin in their farm. So for the distribution of our staph species, we took from the farms, we sampled 274 nasopharyngeal samples, and we were able to recover 212 isolates, of which we were only able to find 126 staphylococci species. And out of these staphylococci species, we found 6 out of 126 were Staphylococcus aureus, 5 out of 126 were Staphylococcus epidermidis, and 59 out of 126 were saprophyticus. And for the ones we couldn't characterize phenotypically, we classified them as other coagulase negative staphylococci, of which were 56 out of 126. Looking at our resistant patterns, we could see that the highest resistance was seen in ampicillin sulbactam, of which 99.2% of our isolates were resistant to, followed by closacillin, 99.2%, cefotaxime, 98.4%, and the least resistance was seen in levofloxacin, of which 1.6% of our isolates were resistant to. So for the multi-drug resistance patterns among the isolates we observed, even though we isolated a small number of Staphylococcus aureus, we found that all Staphylococcus aureus isolates were multi-drug resistant, of which the ones we couldn't speciate phenotypically, the other coagulase negative staphylococci, were 78.6% of them were multi-drug resistant, followed by Staphylococcus saprophyticus, 66.1%, and Staphylococcus epidermidis, 40%. So we also took some data on some farm practices that were going on in these farms. And for these farm practices were the use of antimicrobials, presence of other farms, the visit of other animals within the farm, and antimicrobial prescription, as well as the age of pigs. And we established some associations between these farm practices and multi-drug resistance. And we found that there was a statistical significance between multi-drug resistance and the presence of other farm animals, of which we had a p-value of 0.01, and for other animals in the farm too, there was some statistical significance, of which we found 0.04 of the p-value. So we confirmed that there was a high carriage rate of coagulase negative staphylococci, which is consistent with some research from other global sources, as confirmed by Bonvega in the year 2021 and Chiomba in the year 2025, respectively. So, as Staphylococcus saprophyticus was the dominant species in our study, Bonvega, on the other hand, found Staphylococcus aureus to be the most dominant in their study. We also found a high multi-drug resistance among Staphylococcus saprophyticus, of which were 42.86%, and the other coagulase negative staphylococci, which were 48.35%. And according to Bagava and Zine, the year 2012, this is common among food animals. We also found a statistical link between multi-drug resistance and the presence of other farm animals, indicating that farms housing different types of animals create an environment where resistant bacteria can be shared among these animals. And also we found a link between multi-drug resistance and external farm visits, of which we suggest that animals from external sources could contribute to the spread of resistant bacteria in the farms. Also, we found that the frequent use of antibiotics may result in resistance in some particular drug class, as we saw that most farms used penicillin-based drugs, and for antimicrobial resistant pathogens, the highest resistance was seen in the penicillin-based drugs. So after this, we concluded that there is a high prevalence of coagulase negative staphylococci, especially among Staphylococcus saprophyticus, and a troubling level of multi-drug resistance. Also, we found that multi-drug resistance is not only influenced by antibiotic usage, but also factors like the presence of other farm animals on the farm and the visit of external animals as well. We recommend that further studies employing molecular techniques can be done to speciate other coagulase negative staphylococci further to understand their individual prevalence in pigs of Accra. And also we encourage antimicrobial stewardship among pig farmers in Accra. I also want to take this time to acknowledge some people involved in this study. And first, I want to acknowledge my supervisors, Dr. Richard Mills, Dr. Matoda Imokono, Ms. Rita Ohinilabi, and the pigs farmers involved in our study, and also the entire SecBio UCC team, and the co-authors as well. Thank you.
Thank you, Nathaniel, for sticking to time. You were right on time. Please, do we have any questions for him? Okay. How many? I see 2 hands up already. So we'll take from Dr. Labi. Abana. So someone should come for the mic for them. And then there's a second hand at the back. Yes, please. Thank you so much.
Hello.
Hello.
Okay, so it's about, you gave us the average ages of the pigs you had in this study. Now, I would have been very happy if you had given us an idea of the lifespan, the average lifespan of the pigs.
Okay.
Before we put this into context.
Okay.
My next question, I'll just ask and then you can answer them. My next question is, you said you ended up, all the pigs you recruited were all exotic breeds. Did you specifically aim for that, or they just randomly occurred in the site you chose?
They just randomly occurred.
They just randomly occurred. So in that case, my question to you then is that how would you say, so suppose the study, how do you think that the local species will fare in comparison to the exotic? Okay. And then my last question, you said...
Can I take the questions one more, please?
Oh, well, I want to ask and then I sit down. Okay. I'm sorry about that. So, yes, so you said other animals visiting the farm was a factor.
Yes, please.
I would be very happy if you can elaborate or just expound on that. Does it mean that the pigs were not in enclosed spaces?
Yeah, they were
in what
spaces.
Animals were visiting the farms?
Okay.
Thank you.
Okay, thank you very much for your question. So to answer your first question, you asked about the average age of the animals. So from the animals we sampled, the ages were between 2 months to 6 months. Lifespan. Okay, we didn't actually take background data on the lifespan of the animals involved in our study.
No, you have to know the lifespan of the pig if you are going to utilize it.
Okay. 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 All right.
So at what point are they killed? Like, at what point do you see
Okay, usually...
the pig is matured to be killed?
Usually, from the data we took, we could see that they were mostly killed around the age of 6 months. Okay. So to your second question, please, if you can kindly remind me. If you can kindly remind me of your second question, please.
In comparison to the exotic ones.
Okay, so what I've observed in Ghana is most of the pigs we consume are of exotic breeds, usually. So we mainly focused on those exotic breeds and not comparing them to the local breeds. So we wanted to see the pigs, wanted to see how these pigs could also be susceptible to these resistant strains, but we didn't actually take in consideration the type of exotic breeds, comparing them to the local breeds. We didn't actually take that into consideration. We're mainly focused on these exotic breeds, as we know that most Ghanaians consume these exotic breeds.
Okay, so the other animals visiting the farm?
Okay, so for the other animals visiting the farm, most farms we sampled confirmed the visit of wild birds and even ducks from external sources, not animals from their farms entirely. Even ducks and some domestic animals coming out from different environments onto their farm.
Okay.
Thank you.
Thank you. And then there was one hand at the back.
Yeah, thank you very much. I just want to find out on your sample selection criteria. You said you selected healthy commercial pigs.
Yes, please.
My question is that what do you mean by healthy? Wait. The second question is that if I had found staph aureus in the nose or in the skin, and it's considered as a normal flora, how did you select healthy pigs? What do you mean by healthy pigs? And how would that inform your decision criteria or something?
Okay.
That's my question.
So please let me come clear. So presumably healthy pigs that were not showing any sign of infection on site.
Did you test for that?
No, please.
Thank you, Nathaniel. Please, any further questions? Any further questions? All right, so I might have one question for you.
Okay.
So you said the waste was disposed on the farms.
Yes, ma'am.
How were they doing this? Do they just throw it on the farm, or they will keep it somewhere and then?
Okay. Usually, they dig out, should I say, latrines at the back of the farm, like on a farm site, but outside of the pens. So that's what mostly they were doing.
And has it got anything to do with AMR you found? I mean, has it got any implications for it? And my other question would be, so what is the implication of your study for me who loves eating pork?
All right. Okay. First of all, to answer your question, we do know that Staphylococcus saprophyticus is a gut microbiota, and mostly we do not find them in the nasopharynx of pigs. So,
papillomavirus infection, factors influencing uptake of screening by commercial sex workers in 2 municipalities of Ghana. Okay, thank you.
Hello, everyone. I stand on already existing protocol to present my research on human papillomavirus. Before I begin, I would like to share a brief experience on my field work. When I started collecting data, I realized that to truly understand the world of commercial sex workers, I had to step in their shoes, at least. Yes. If I mean step in their shoes, at least in perception. There were moments where I had to blend in to listen without charging them. I entered into spaces to earn trust, spaces where researchers were hardly welcomed. It was then I realized that data is not just numbers, it's lived experience. This insight shaped the heart of my study.
So could someone come and help us Someone from the tech, IT.
My presentation will be taken through from the background through to the summary. Cervical cancer remains one of the leading causes of cancer-related death among women. Human papillomavirus, HPV, is the main risk factor of cervical cancer. HPV is commonly found among females with multiple sex partners, particularly commercial sex workers. Screening has been found to be the primary method and cost-effective way to prevent cancer, cervical cancer. However, previous studies showed that commercial sex workers have low knowledge of HPV, and the uptake of HPV screening among this group is low. Therefore, no known studies have been conducted among commercial sex workers in the Ho and Hokwe municipalities which sought to explore the factors influencing uptake of HPV screening. It is therefore necessary to identify individual, social, and health system-related barriers to the uptake of HPV screening in these municipalities. Hence, the aim of my study explored the factors influencing the uptake of HPV screening by commercial sex workers in these two municipalities of the Volta region. My specific objectives were determine the level of uptake of HPV screening, explore the individual level factors, explore the health system related factors, and then explore social factors that influence HPV screening. My study used an explanatory exploratory study design. The study was situated in Ho and Hokwe municipalities. My study included female commercial sex workers. My study population comprises of female commercial sex workers in Ho and Hokwe municipalities. The study excluded female commercial sex workers who were not permanent residents of Ho and Hokwe but were around on temporary visits. The study used a snowballing sampling procedure, a quantitative study of 60 commercial sex workers, and the eminent issues were explored using 7 in-depth interviews. Quantitatively, a pre-tested semi-structured questionnaire was used, and qualitatively, we used an in-depth interview guide alongside handwritten notes and an audio recorder. Ethical clearance was sought from the UHAS Research and Ethics Committee. All participants provided informed consent. We ensured privacy and confidentiality by allocating code names. Quantitative data was used descriptively using Stata version 17. Codes and teams were developed thematically using Atlas TI version 9. Our study found that 96% of commercial sex workers have never had any form of HPV screening, whereas in terms of HPV uptake by municipality, 10% of commercial sex workers in Hokwe have taken some form of HPV screening, whilst not even a single commercial sex worker in Ho has ever taken some form of HPV screening. Before I move to the next step, it also emerged on field that HPV knowledge among commercial sex workers remain very low. To our qualitative findings, we had 3 sub-teams, we had 3 teams emerging as barriers. We had 3 teams emerging. Whilst under the first team, individual factors, we had 3 sub-teams emerging as barriers. That's lack of knowledge about the screening, inability to afford screening, and fear of pain. This is what a commercial sex worker had to say. The reason why I didn't do the screening is because I don't know about it. And I don't know how important it is for me. I don't even know if it is painful or not. Commercial sex worker 4 had to say this. If I don't have money and I cannot afford the screening, I will not be able to do it. I won't kill myself. 2 sub-teams emerged as facilitators or the motivators. Education about the screening and less painful screening. Commercial sex worker 1 said, maybe more education and awareness creation on screening, maybe on radio or TV, and people going around to educate us on what HPV is. It will enable us and push us to go for the screening. The second team, social factor, we had 2 sub-teams emerging as barriers. That's peers or friends not taking the HPV screening and stigma by community members. Commercial sex worker 2 said, if you are going to check yourself for HIV or screening, they will think because if you are having sex, that is why you are checking. They will think because you are having sex, that is why you are checking. For me, I will not do the screening in my community. Also, because our religion says we should not do commercial sex work, people will not do the screening because they will be scared of what people will say when they see them doing the screening. We had one sub-teen emerging as a facilitator. That's friends getting screened. If my friends or co-workers go and do the screening and they say it is normal and safe, I will also go and do it. The third team, health system related factor, we had 2 sub-teams emerging as barriers. That's distance to health facility and non-availability of HPV screening. The screening is not that popular where I stay, but if it is available here, I will go and do it, says commercial sex worker 5. We had screening services made available to them and made free. Positive attitude of care workers towards commercial sex workers was also noted, and if recommended by a health worker. If the people, that's the health professionals doing the screening, are not nice, I won't go and do the screening. But if they are nice people, I will surely go and do it. Our study found that uptake of HPV among commercial sex workers were poor. What does this imply on public health? Commercial sex workers will be more prone to the acquisition and transmission of HPV. Also, our study found that about a half of commercial sex workers do not have any knowledge on HPV infection. Also, similar studies in Nigeria by Shu et al. Found that slightly a half of commercial sex workers had a good HPV knowledge. Commercial sex also, a major barrier was financial constraints, where commercial sex workers' economic and financial status serve as barrier to the uptake of HPV screening. Previous studies also found that women with weaker health purchasing power are less likely to uptake sexual and reproductive health. Therefore, based on our study, I recommend that health authorities, more specifically the Public Health Department, should organize regular educational and sensitization programs that are targeted towards this vulnerable group. Also, the Ho and Hokwe Municipal Health Directorate, specifically the Health Promotion Unit, should develop a systematic sensitization program on what is involved in the HPV screening process and the number of times to be screened to address some fears by clients who find the process a mystery. Finally, the good news is the Ghana Health Service should also regularly organize in-service training and workshop to reorient healthcare providers. In summary, knowledge and uptake of HPV screening among commercial sex workers were poor. Several barriers emerged, such as inability to afford screening, fear of pain, and lack of knowledge were some of the barriers that hindered HPV screening uptake among commercial sex workers. Therefore, innovative interventions are needed to promote HPV screening among this group. The good news is Ghana has introduced HPV vaccine for children between ages 9 to 14, which took place in October. Most likely next year, we hope high-risk groups such as commercial sex workers who are not already infected should be targeted. My references, thank you.
Okay, thank you, Dr. Timbisa, for a really nice talk. So now we want questions or comments from the floor. Okay, so, yes, sir.
All right, thank you. My name is Kenneth Oswajiman. I want to know about your work. It's quite complex. How did you enter? Through their leader or what? And also, secondly, what did you deal with? Is it those who are stationary or those who are roaming in your study? All right, thank you.
Okay, so in research, we have something called a sampling procedure, which I mentioned in my study, the snowballing procedure or technique. Yeah, so I had to identify hotspot areas where this group of people work, especially the pubs. I had to rely on pub owners to recruit commercial sex workers, and then I depend on the first commercial sex worker to link me to.
Yes, thanks for the nice presentation. You indicated targeted
education for them. From the question that came, how do you target them? Okay. Because people are, don't want to be exposed or don't want to be seen. Okay. So how do you ensure that targeted?
Okay. It was not an easy experience, I'll mention. So getting them was really tedious. When I relied on the gatekeeper, which is a pub owner, to get me one person. But then getting them to come out of their shell was really a bit of an issue. So I had to go off the line of research to, please them in the way where they can come out. So pleasing them, I had to rely on buying weeds, amnesia, alcohol, things that will bring them on board, because these are the kind of things they involve in. So that's a bit of a challenge.
Okay, yeah.
Okay, so.
Okay, next question.
Okay, so my question had to do with pain came up a number of times.
Yes.
Okay, now, did they explain what kind of pain they feared? Being commercial sex workers, one would have thought that they'll be used to pain.
So it all has to do with the knowledge of what HPV screening is. It's because they had no knowledge of what HPV is, what goes into it. They tend to believe that the HPV screening will be painful, so they are scared.
Okay.
They may get hurt in the process, even though they are used to pain.
Okay. May I have a related question and comment? Yeah, two brief questions. One is, sorry, maybe we missed it. How many number of workers did you outreach?
60 commercial sex workers.
Yeah, right. Okay,
yes.
Thank you. And another thing is, what to add for the details of outreaching and persuading, the increase of tests on HPV infection. So do you have any ideas? Because it's also an issue in Japan because we didn't have regular vaccination until quite recently, and how to persuade amongst the really the limited number but highlighted adverse cases. It's really kind of a controversy right now for us. Do you have any ideas?
Ideas
of what would be a persuading detail or hook to request them, or do
yeah,
these kind of
I
tests?
don't really get you.
Oh, sorry. I'm really sorry. Yeah,
what did you tell them?
What did I tell them? Oh, okay. Yeah, sure.
Okay, so let me rephrase then. For example, we have different serotypes.
Yes.
Right, for papillomavirus. Some are rather benign, and some are quite aggressive. Yeah. So I'm wondering how this could be compressed as a detail to kind of inform them. If you just say HPV, the word alone, some people might throw it away instantly.
Yes. I had to explain to them what goes in.
Right.
Most people mistake HPV and cervical cancer for breast cancer. Yes, it came up. I asked you in my in-depth interviews, my questions. I asked you, do you know human papilloma virus? No. Do you know cervical cancer? No. Do you know breast cancer? Yes. There were instances where I asked one commercial sex worker what cervical cancer is, and she said it's the cancer of the breast.
Okay. So maybe kind of normalizing, understanding of that part might be the preceding step.
Yes. Important. So we had to go down, break it down. It was not so complex. The questions were not complex.
Okay, forget this. Okay, it's normal, anatomical.
Yes, anatomical.
Okay, thank you very much. Okay, so we're about time. Thank you, Dr. Timpisa. Thank you.
Hello.
Yeah. One more question?
Yes. Final person, please.
Okay, so I think I'm on the floor. I am very curious. I am quite curious by your own words. I had to please them, quote and unquote.
That's why I said I had to.
Let me land,
of
please.
Research.
And I am
curious with this because a colleague of mine was also doing some investigation in HIV, and he was a man. Apparently, by our own engagement with him at our level, informal, he told us that, look, at a point, I have to pretend as if I was one of their clients.
Yes.
Because they refused to engage with me, and I needed the data for research. And borders on ethics, if this is submitted for a paper, and I'm sure you use pleased, I'm sure you know of coercion.
Yeah.
Please, how do you deal with this?
I use please in the sense where, you had to blend in with them. So there were I remember when I started, I started dressing like a professional going, and one of their bosses told me, Madam, if you really want my girls to talk to you, please start acting like us. So I had to go to the pub one night, dressed like them. I went with condoms to display condoms. Excuse my words. We are all public health people here, so use condoms to display. I displayed condoms. Oh yes, like this is what I do. Like come for the night, we are doing this. That is what I meant by please in that sense. And I was dealing with female commercial sex workers, so please is not what you are thinking.
All right,
Okay,
thank you so much. And thank you for the discussions.
Thank you. Okay, now I pass over.
All right. So our next talk is from Jonathan Kluche. Jonathan is from biology department. He works with the HIV group. And his presentation today is latent TB infection modulates T-cell exhaustion in people with HIV on antiretroviral therapy. All right, all the best.
Thank you. Good afternoon, all. So as already mentioned, I'm Jonathan Kluche. I'm a research assistant with the HIV Cure Research and Infrastructural Study team here at the Noguchi Memorial Institute for Medical Research. And standing on already established protocol, I'll be presenting one of our projects that looked at the impact, how latent TB impacts T-cell exhaustion in people living with HIV. Since its first discovery in the early 1980s, HIV continues to be a serious public health challenge, with over 40 million people living with HIV as of 2024. And it's rather unfortunate, Africa bears a proportionate burden of HIV with over 26.3 million people living with HIV. That's about 65% of the global population of people with HIV. By the end of last year, 77% of the total number of people living with HIV were receiving antiretroviral therapy. Whilst ART has proven efficacy for long-term suppression of HIV-1, it sadly still does not provide a definitive cure. So during the early stage of HIV infection, there is actually high viral load, as we can see in the graph. But once ART is initiated, there is decline in viral load when you take the person's sample and test, and it gets to a point that you test and the virus is undetected. And at this stage, the virus can also not be transmitted. But sadly, once treatment is interrupted, we observe viral rebound. What this means for people living with HIV is that they need to commit to lifelong antiretroviral therapy in order to suppress the virus and also prevent the transition into AIDS. Now, the major challenge to an HIV cure is the latent reservoir, which we refer to as the sleeper cells. So these are basically immune cells, that's the CD4 and macrophages, that harbor the latent provirus in a transcriptionally silent state. So when you look at HIV infection, HIV usually infects CD4 T cell, able to integrate its genome into the genome of the host, replicate, and as the immune system undergo its own course, the immune system is able to clear some of the infected cells. But for taking memory of infection, some of these infected CD4 T cells go into latency, keeping memory of infection. HIV is also known to directly infect resident memory CD4 T cells. And since these cells are not actively transcribing, they harbor the latent provirus in their genome.
And other mechanisms such as epigenetic changes, transcriptional factors, and immune metabolism also contribute to the latent reservoir. Now, Africa holds a rich and diverse repository of microbial life, of which TB is not an exception. And with over 2.5 million cases of TB, over a third of them occur among people living with HIV. And TB is the leading cause of morbidity and mortality among people with HIV. Studies have shown that co-infections such as latent TB usually produce antigens that contribute to the persistent immune activation during HIV infection. And since latent TB reside in granulomas, residual antigen production from these granulomas may result in viral seeding of more CD4 T cells, which could potentially increase the reservoir size. So this laid our scientific premise for this study, where we sought to identify the differences in T cell responses, look at the HIV reservoir size between HIV only and HIV latent TB patients. Specifically, we determined the impact of latent tuberculosis infection on the differential secretion of immune activation and exhaustion markers, cytokine production, and the HIV reservoir size between HIV only and HIV latent TB patients. Now, so to do this work, we first of all screened a cohort of 390 individuals. So these are people living with HIV. This cohort was established back in 2019 that we've been following them up every 6 months till date. So we screened them for latent tuberculosis using the interferon gamma release assay. And we found that 37, that's 9.5% of them have latent tuberculosis infection. So in the lab, what did we do? First, we collected 100 ml of whole blood from these participants. So the positive individuals for latent TB were matched based on age and gender. So we collected 100 ml of blood, from which we isolated PBMCs, and from this PBMC, we isolated total CD4 and CD8 T cells. The T cells were stimulated for a period of 19 hours. And in step 2, we seeded the stimulated cells and stained them with antibodies specific to immune activation and exhaustion markers and acquired and analyzed using flow cytometry. For step 3, we determined the cytokine profile of our study participants using luminous multiplex assay. And for the HIV reservoir size and latency reversing capacity, we use qPCR. Now, our results show high expression of both immune activation and exhaustion markers. For immune activation markers, we are looking at high expression of CD25, CD38, CD69, and HLA-DR being the main drivers of persistent immune activation in HIV latent TB patients compared to HIV monoinfection. For exhaustion, we noticed PD1, LAC3, and CD244 were the driving markers of T cell exhaustion. For cytokine production, we noticed that in latent TB co-infected patients, there was high production of interferon gamma, TNF alpha, interferon alpha, IL-17, IL-1 beta, IL-6, and IL-10. And usually, we should expect low production of these cytokines. But because of latent TB infection, these cytokines were upregulated to keep latent TB in its latent state. We also observe a reduced production of interleukin-1 beta, interleukin-6, interleukin-10, 17 in HIV latent TB patients compared to their mono-infected counterpart. And once there is reduced production of these cytokines, there is an indication that these CD8 T cells are exhausted. And in the exhausted state, the CD8 T cells, which are cytotoxic T cells, will not be able to kill infected CD4 T cells in the immune system, thereby potentially increasing the reservoir size of HIV, which is the major challenge to an HIV cure. Now, how does these findings translate to cure strategies that are being developed? So we looked at how we'll be able to reactivate the latent provirus using known epigenetic modifying compounds, as well as some novel natural products that we are screening in our lab. And as you can see, it is easier to reactivate the latent provirus in individuals with HIV monoinfection compared to those infected with latent TB. Now, how does all these findings translate into the reservoir size of the study participants? So we observe higher levels of unspliced RNA, which is an indication of active transcription due to the persistent stimulation of T cells by residual antigens from latent TB. And we also observe that in co-infected patients, there is higher reservoir size in terms of the proviral DNA copies that we are seeing. In summary, our study has shown that there is expression of immune activation and exhaustion markers with CD38 and CD69 being the main drivers of immune activation during HIV-TB co-infection. We also observe larger reservoir size in HIV latent TB co-infection compared to mono-infection. And this has contributed to the low latency reversing potential observed in the co-infected individuals. Our findings suggest that there is a need for a cure strategy that is targeted for co-infection in HIV infection. Here are a few references. I'd like to acknowledge the massive action team that made it possible for this work, and also our collaborators back here in the institute and outside the university, and also our funders. Thank you also for your time and listening.
Thank you for that very nice and insightful presentation. Please, do we have any questions for him? Yes, please.
Thank you very much for the presentation. I just want some clarification on your controls. You did mention that you were taking TB infected people who were latent, and then they also had HIV, and then they were on ART. Am I correct? Yes, please, you are correct. Okay, so were you looking at people that, they've been on ART for more than 1 year, and then they were at an undetectable stage? Or you were looking at people that they had a HIV, but then, they had levels of viral load that were detectable?
Thank you for that excellent question. So for this study, we did matching, so case control matching. So we took notice of years they've been on ART in doing the matching. And also, all these participants are virologically suppressed. So they have undetectable HIV mRNA using GeneSpec.
The next one is, we have different ARTs. And what a doctor may give to this particular HIV-infected person may be different from what another person may also receive. How did you rule out the fact that it could be that an ART is contributing to the exhaustion markers that you are actually seeing, or they are sort of having an effect on what you are trying to measure, the outcome you are trying to measure? Did you sort of stratify, okay, we have a group that they are taking ART-A, and then we have another group that they are taking ART-B. Do you get the point? But then it wouldn't be like you are analyzing based on people that they are taking ART-A and B, and then you're also analyzing people that they are taking ART-A and C, that kind of messed up. So that you sort of, when you get that stratification, you'll be able to tell that, you'll be able to control for the effect of ART on what you are trying to measure.
Thank you very much. So in our matching, as I mentioned, we started following up these patients since 2019. So we have clinical data on the ARTs they are on. So in our matching, we make sure the people we are picking are on the same ART. And just a point of education, ART will help suppress viral replication, not exhaustion, the expression of exhaustion or activation markers. Thank you very much. Yeah, but there is the possibility that, it's a factor. And as you are doing, you have to control for it. So since you have your test samples, you should just make sure you have things normalized out. But then aside that, I wanted to ask, did you consider the fact of using a healthy patient, like someone that does not have HIV at all, or someone that just have HIV but not on ART? Thank you. So the study also recruited healthy participants, matched based on age and gender. Thank you very much.
Okay, but then why did you do that?
So one of the clinical defining features of HIV is immune dysregulation. So to rectify that, what we are saying is, as a result of latent TB or HIV, we need to bring on healthy donors, which we know their immune system is functioning properly.
Yeah, so you normalize for that one. Just like I was saying for ART, just so you normalize across so that you just know it's not ART that may be contributing to the outcome you are measuring. Thank you.
All right, thank you very much.
Thank you so much. Any further questions? Wow, okay, 3 more. So there's one at the back, one here in the middle, and then Prof. Ampofu. Yes, please. So let's start from the back. I saw him first.
Thank you for your presentation. And then the question I wanted to ask is a bit of clarification. I think you said you were measuring the interferon gamma responses to screen for those who had the TB, right? I wanted to know, interferon gamma is kind of like general for most infections, like immune responses. So was this specific for TB, or it was the general responses?
All right, thank you very much. So interferon gamma is one of the cytokines that is needed to keep TB in the latent state. So the kit we use, which is the quantiferon, uses interferon gamma, and it has a specificity of about 90%. So this interferon was specific to latent TB. Thank you.
All right, yes, the next one.
That was a great presentation. I just wanted to ask, I'm just curious if in terms with the phenotypes that you looked at, the markers, exhaustion markers, were you able to also look at how these markers are associated with HIV genotypes? I don't know if you have data on that. It would be interesting to see how those sort of genotypes correlate with the phenotype markers you are seeing in terms of the immune markers.
All right, thank you very much. So this is an ongoing project, and the question you just asked is being looked at in another study. Thank you very much.
All right, then we have Prof. Ampofu. Please come forward. Bring the microphone.
Thank you for a very well-structured presentation. I was impressed with your knowledge of the subject. You didn't tell us whether, you said TB latency, but you talked about ART treatment. I want to know about TB treatment, the history of TB treatment because you talked about, you matched them in terms of age and then also HIV, TB latency. But what about TB treatment?
All right, Prof, thank you very much for that excellent question. So for TB treatment, we've not taken that into consideration, so it's something that we'll take up further and explore in the cohorts. Thank you very much.
Thank you, Prof. Jonathan, I hope you know the question I'm going to ask. Do you know? You should know, because I've asked this several times. What does this mean for HIV cure?
So we are getting close to a cure, and for what it means for an HIV cure, we need to focus on drugs that will be able to have the ability to activate the latent provirus in co-infections, not just limited to latent TB, but other co-infections such as HPV and HBV co-infection. Thank you very much.
Thank you so much. Let's give Jonathan. And then we'll move on to our next presentation.
Okay, so the next presenter will be Dr. Joycelyn Dame. Yes.
Let's calm down.
And her talk is going to be antibiotic use in respiratory syncytial virus positive children under 2 years, a cross-sectional study at a tertiary hospital in Ghana. Thank you very much. Yeah.
Thank you very much, and good afternoon to you all. I stand on all existing protocols. My name is Joycelyn Dame. I'm a clinician. I'm an infectious disease specialist at the Kolebu Teaching Hospital. So this presentation will be a little different from what you've already been seeing. I make this presentation on behalf of my co-authors. So our sponsors for this project was from UMC Utrecht, and also the subsiding party was the Bill and Melinda Gates Foundation. So I'll start with a general summary of what this presentation is all about. What was the question that we sought to find an answer to? We sought to find out the frequency and clinical determinants of antibiotic use among RSV-positive children under 2 years. What did we find? Majority of these children who had an RSV infection received antibiotics. And clinical determinants that were predictors was tachypnea. Tachypnea is when you find you're breathing very fast, and difficulty in breathing were predictors of antibiotic use. But why is this important to me? Why is it important to all of us here? Because using antibiotics for a viral infection promotes antimicrobial resistance. This means that we need antimicrobial stewardship, especially in pediatric care. We need preventive strategies, such as the RSV vaccination as well. So for the background, RSV is a virus, a single-stranded virus. Is a leading cause of acute respiratory infections in children of all age groups, from the young to the very old. And it causes various clinical symptoms. Some are mild and some may be severe. Now, when it is severe, it can actually overlap with pneumonia, which is a bacterial infection. And so it is difficult to tell that, is this RSV a virus or is this bacteria? In places where there is advanced technology, when a child like this appears, what you do is diagnostic, a viral panel will be done, and you can determine whether this is virus, and so I can stop my antibiotics and not promote resistance and just give supportive therapy. But in settings where this is not available, or even when available, expensive, we are not able to do that. So what was our study objective? We wanted to evaluate antibiotic use and its clinical determinants in children with RSV-positive illness under 2 years, presenting at the Department of Child Health, where I work, in the Kolebu Teaching Hospital. Now, this study, which we did with antibiotics, was actually nested within a larger prospective observational study known as the RSV Goal 3 Health Economic Study. That study was looking at the direct and indirect cost associated with RSV infection in the same age group. It was an international multi-center study, and the Ghanaian arm was at the Department of Child Health, pictured there. It was in children less than 2, as I already said, and the duration of study was from June to November. Why? Because this is the period where studies had shown that this RSV seasonality occurs in Ghana. Who did we include? 2 groups, inpatients and outpatients. For inpatients, we admitted those who had severe acute respiratory infection. That was in line with WHO's extended definition, and that is any child who presents with an acute respiratory infection, either cough, difficulty in breathing, within an onset of 10 days. So for those who are severe, those who are admitted are classified as severe, and outpatients are non-severe. We excluded those less than 4 days. And the reason is that these are babies who have just been born and could have had other problems, such as, we call something meconium aspiration or even early neonatal sepsis, and it will be difficult to be able to differentiate it from an RSV infection. Also, if the child came in less than 2 years and you were hospitalized for another reason, of course we did not recruit you. Or if for any reason, we know the reason of your respiratory distress, such as you can have a cardiac disease, so you're in cardiac failure, your heart is not working well, and so we know that that is why you're having difficulty in breathing, we will not recruit you. So what were the methods for diagnosis and data collection? RSV was tested using a point-of-care test. This kit has been previously validated in a previous study in the same site. And also, we did reverse transcriptase PCR testing of all samples. So every sample that would use the point-of-care test would also send it to Noguchi right here to confirm the presence of RSV. and a test was considered positive either by PCO testing or by PCR. So we collected variables such as the age, the sex, underlying medical conditions. Something like prematurity is associated with severe RSV infection. The clinical presentation, how they came in, difficulty in breathing, fever, those who needed oxygen, oxygen at admission, etc. Then also the type of antibiotics that they were put on, and the number of antibiotics, because some people would even have more than one antibiotic. For the analysis, we made comparisons between RSV positive patients who received antibiotics and those who did not receive. We also categorized the antibiotic according to the WHO aware system, where it is categorized as in access antibiotics. Access are the antibiotics that do not promote resistance. They are more narrow spectrum, and WHO recommends that at least every country, you should have at least about 70% of all your antibiotic use should be from the access. The watch group tend to promote resistance a bit, so they are more reserved. And so it's when you have maybe an infection with a drug-resistant organism or severe infection, then you would use them. And the reserve, as the name is, antibiotics that are really reserved. Don't use it like polymyxin, colistin, when it is when you really need it. We conducted a logistic regression to predict the use of antibiotics and considered a p-value of less than 0.05 significant. So the total number of all participants, majority of them were RSV positive, that is 56%. And these are the patients in whom we looked at their antibiotic use. So what was interesting to note is that even though factors such as median age, sex, etc. were not significant, the clinical determinants, those who came in with tachypnea, that is breathing fast, those who needed oxygen, and those who were breathing with much difficulty, they were the ones more likely to have antibiotics used. On regression, again, the predictors were those who had difficulty in breathing and tachypnea. These continue to be significant. So it shows that if the person came in more ill, it's difficult to tell that this is bacterial or it is a virus. So we would start with antibiotics because I don't know, I don't want any child to die, etc. But what were the type of antibiotics that were used? And of much concern for me is the fact that third generation and beta-lactam inhibitors, such as amoxiclav, these are antibiotics that are more broad. They were the second most frequent antibiotic use. Happy to say that at least the most frequent used were penicillins. And so majority were coming from the access groups. However, these are children who had a virus infection. Majority, again, were found from the access group. And there were a group of children who also had co-infections and no co-infections. Among those with the no co-infections, the common diagnoses were a pneumonia, upper respiratory tract infection, or bronchiolitis, bearing in mind that all this is RSV infection. So we noted that there was majority use of antibiotics among children with viral infections. And what this means is that using these antibiotics, we are promoting resistance. There's an urgent need for stewardship, especially diagnostic stewardship. A child has come in, it's okay for us, we should be able to do viral panels. We should be able to do tests that will determine whether it is virus or bacteria. And also we should look at RSV. We recommend that in line with WHO's current position paper on immunization on RSV, I think Ghana needs to take a look at it. We recommend that pregnant women in their third trimester receive RSV vaccine to protect the baby for at least 6 months. That is the period when you have the RSV infection. Of course, there were some limitations. We did not routinely test for bacteria. We did not explore the rationale behind antibiotic prescribing. It was a single center study. We cannot extrapolate it to other centers, and we did not look at long-term outcomes. But again, we conclude that there was still a high level of antibiotic that was used in these children, promoting resistance, antimicrobial resistance. And so there's an urgent need for stewardship, antimicrobial stewardship, especially in pediatric care, and we need to look at diagnostic stewardship more closely. I acknowledge all the children and parents who agreed, and also the team. This was the study team involved, and I'm happy to take your questions and comments.
Thank you, Dr. Nami, for a really important. Okay, so we have hands from the front. Yeah, front. Yeah, yeah. Yep. Then the second class.
Thank you. Sorry. Okay, so my first question is about the assessment of infection. So you said if it was either POC positive or the confirmation test.
Or, yes. And or.
Yes. And or.
Yes.
Okay, so did you have a lot of both positive for both
Majority.
cases?
Majority were, I didn't show the data, but majority were confirmed positive by both. There were just a few that were just PCR positive, but we considered all of them as positive as well.
Now, looking at the model, I noticed most of the confidence intervals were really wide, super wide.
Yeah.
So just because of that, I just want to ask about the sample size estimation. How did you estimate your sample size?
Okay, so because this was a study that was nested within the larger study, their sample size was about 148, but the total number of participants I think we had was 128, and that is what we used.
The total number of participants?
The final number that we use. So the sample size had been determined by the initial study.
By the initial one?
But yes.
But not yours?
But not yours. We use the same population. But for their sample size, a previous study where they did RSV ICU study, they used that the numbers of RSV patients to calculate their sample size, which came to almost 150, I think, 148, 150. But the final sample size, I didn't show that they are working.
Okay. So based on that, if you can just briefly show the model again, based on the logistic regression model you had over there.
They have to show it at the back.
Tech. IT. She needs to go back to her slides.
Yeah. Slides again, please.
Yeah, it's coming up.
Okay. Back the model. Yeah. Okay. So briefing difficulty. So let's say 4.2 to 263.
They were really wide. Yes.
Really wide, yeah, extremely really wide.
And I think it's because of, again, the sample size was quite small, because just among those within who had the infection, there was a smaller number of all the participants. Those who were used antibiotics, it was a smaller number.
Okay.
So that is just 72.
So in the context of that, how would you interpret your findings?
I still think it's very important, the fact that we are, because like I said, clinically, the fact that we are using antibiotics among this group who we know have an RSV infection, it's important. And if this is because of the, we had a study and we had the point of care test, it's something that is not done routinely. So these are children that it happens all the time. But we were able to stop antibiotics in this group because we knew that it was an RSV infection and give supportive care. So I still think it is important.
Okay, true. So, but then we would at some point need a larger sample size.
Absolutely.
And then having the extremely wide confidence intervals makes the estimates unstable.
Sure.
So that we just have to interpret them with some amount of caution.
Absolutely.
Thank you.
Okay.
Thank you. Okay, we have hands in the middle, and then, yeah, two. Okay, we have two more then.
Right. Good afternoon, everyone.
Hello.
That's a very interesting study, and without any doubt, you're also a fantastic presenter. Thank you very much. My comments or questions goes those. We are in the lab, and one of the major challenges that we have with practice of medicine in low and middle income countries is on the utility of the laboratory.
Absolutely.
Yes, we are trusting science so that we can have improved diagnostics, points of care, but even the ones we have, we are so worried, and it's saddening because we have been deprived of opportunity to have reliable data because we only get tip of the icebergs of those patients' samples being sent to the lab. This study, for example, these patients were tachypnic, and there is no single doctor on earth that will not give them antibiotics. Absolutely. Because there is no indication that this cannot be bacteria. Yeah. However, we would have been able to reduce or eliminate confounding bacterial infections if just at least one sample of blood culture was taken. And this would have increased the value of this study exponentially. For example, we can't say that this result or these patients only had RSVs because they don't have bacterial blood culture that could talk about sepsis. Tachypnea, they could be septic. That is one. And then on the other hand, we know we have challenges producing sputum because they are babies. Yeah. But we could do bronchoalveolar lavage, we could do all those other things. But at least with fever, tachypnea, indicative of sepsis, we would have thought that they would have taken blood culture along the line of management, even if there was no research. Then as a research, you would have thought that you go, maybe you did, you just didn't report it. But I would like to know why you didn't do that, if you didn't do. Then number two, mortality, because these were really bad cases, so to say. So I would have loved to know what is the rate of mortality in the group that used antibiotics and the group that didn't use antibiotics. And then maybe you would have been able to say maybe, oh, after all, the antibiotics were justified. Even though we didn't have laboratory basis, but at least the end sometimes justifies the means. We would have liked to know that. Maybe I should just leave it at this too. Thank you very much.
Thank you.
Thank you. And one more question.
Okay, so I'll just respond. Yeah. And so what you said initially about blood cultures is very true. And this was not done just because it was not part of the study. And I must say that unfortunately, routine blood cultures are not done because they are not in health insurance. And it costs almost 200 Ghana cedis to do a blood culture. And I work in a setting where my patients are a little deprived. So except currently I'm doing a study where we are just looking at blood cultures, and that's because we are funding it. But once it doesn't come from funding, it's something that doesn't happen often, which is unfortunate. I agree with you. And that's why we say that it was one of our major limitations, that it wasn't. Concerning mortality, there was one mortality, and that was a child who actually had another co-diagnosis. It was a child who had what we call a gastroschisis. It was a congenital, another kind of infection, disease. So it was a co-infection, but it was not what we thought was associated with either use of antibiotics or not. But again, it was a much smaller sample, that's why.
Okay, and the final question, yes, sir?
All right, thank you so much for your presentation. As a next step, you mentioned diagnostic stewardship.
Yeah.
What do you actually mean? Can you throw more light on it so that we can actually translate it into maybe clinical care as well?
Absolutely. Okay, so I love that question because I've started a program on antimicrobial stewardship, and antimicrobial stewardship has to do with the practice of ensuring that antibiotics are being used at the right time for the right patients and for the right need, okay, at the right dose, all the rights, and that is what stewardship is about. When you add diagnostic stewardship, it's being able to implement your diagnostics. So as she already mentioned, you go to a patient and they've started antibiotics on this patient. You would like to know, has a blood culture been done? The importance of that blood culture is to make sure that you can isolate an organism, and if you do isolate, you can either tailor down your antibiotics, because usually you would start broad, and then so that is one aspect, being able to just do cultures, and you use the cultures to influence the use of your antibiotics. Other diagnostics, like with this study, is what we call a respiratory viral panel. I think it's even been done here in Noguchi, where you can have a host of viruses. A child will come in with difficulty in breathing. You do a host of viruses, and then you can tell that this is coming from this kind of infection. So that is also diagnostic. So the child will come in, yes, the child has difficulty in breathing, is coughing, is severely ill. it makes sense to start with your antibiotics. But after you have done your diagnostics, then you can tell that this is actually a virus. Because again, you look at the setting, we have the RSV season, you have the type of patients, you can determine that what we have right now is actually the RSV. And like I also mentioned, RSV can cause severe illness, and what they would need is oxygen support. So again, diagnostic stewardship with the fact that you collaborate with the lab will help you. If you want to do so, I'll give an example of just briefly of even my own son who had, he came home and I think there was a flu A season had occurred. And so very febrile, high temperatures, coughing, some kind of difficulty in breathing a bit, but it did not need oxygen. I was able to do the respiratory viral panel. It was flu A, and I got Tamiflu, and I used it. That is using diagnostic stewardship, and antibiotics were stopped immediately. But that test cost 850 cedis. How many people are going to be able to afford that? Okay, so that is when we need to collaborate more with our labs to make sure the prices of these things come down, and every child has access to diagnostics. Thank you. I hope that answers your question.
Okay, thank you, Jocelyn, for a wonderful talk. Again, a passing over.
Thank you, Doc. Please, we will talk after this session. I'm wondering how these things happen, but we'll talk after the session. Thank you. So our next presenter is Diana Atandem. She is also from the virology department, and she will present to us on polymorphisms in the matrix metalloproteinase 3 gene are associated with hepatitis B virus infection in Ghanaian adults. Thank you, Dinah.
Okay, hello everyone. So I'm going to talk about findings from our study titled Polymorphisms in the matrix metalloproteinase 3 gene are associated with hepatitis B virus infection in Ghanaian adults. So hepatitis B is still a major public health threat, and over 200 million people live with the infection worldwide, and every year over 1 million new infections are recorded with an equally matched number of deaths. Africa bears a disproportionate brunt of the burden, with more than 60 million people living with chronic Hep B in the continent. And the situation is not different in Ghana. In Ghana, there's close to 3 million people with chronic Hep B, and only less than 1% gets diagnosed. And Dr. Yvonne's group did a hospital study that suggests that as you are moving from the south to the north, the prevalence increases. Hepatitis B virus targets the liver mainly, and once the virus gets into the liver, it attaches to the liver cells, and although it doesn't lyse the cells, it changes the cell's morphology, leading to the production of reactive oxygen species and DAMs. And these trigger a cascade of immune responses which lead to the establishment of inflammation, and through these processes scar tissue is deposited in the liver, and that is what is usually termed cirrhosis, and then as it progresses it can move to fibrosis and then sometimes cancer. Now in a normal liver, matrix metalloproteinases will come in, degrade the scar tissue so that the liver can regenerate. But when there's a problem, this ECM breakdown can be impaired. Now, matrix metalloproteinases are a group of 23 zinc-dependent enzymes, and they have diverse functions, and the main one is to break down extracellular matrix. MMP3 in particular is constitutively expressed in the liver. It is involved in hepatic fibrogenesis, especially during chronic viral hepatitis, and it's also able to activate other matrix metalloproteinases to help with scar tissue degradation. And studies have shown that in different stages of liver injury, there are elevated levels of MMP3. However, we do not know much about the genetic impact or the genetic influence of these, the MMP3 genes on these processes. So this study sought to examine the full-length MMP3 gene in a Ghanaian cohort of HBV-infected people to find out how it influences HBV infection and whether it has any impact at all on liver health. So the main objective was first to successfully sequence the full-length gene and then to interrogate the gene for polymorphisms that could influence HBV infection. This study is part of a larger study that was studying the clinical and immunopathological consequences of Hep B and malaria co-infection. Participants were recruited from both Kolebu Teaching Hospital and the University Hospital at Legon. And we recruited cases, that is, people that are living with chronic hepatitis B but were not on treatments, meaning they had viral loads of 2,000 IU per ml or lower. And controls were healthy individuals who didn't have any evidence of liver infection. The studies were approved by the Noguchi IRB and the Ethics Committee at Kolebu. And all patients gave us informed consents before data collection. So what we did was to collect blood from each person, centrifuge the blood to obtain serum, and from the serum samples we tested for liver markers, ALT, AST, ALP, and LDH. And then from the same serum we extracted DNA, did a full-length amplification of the MMP3 gene, and then prepared libraries from the amplicons. The libraries were then sequenced on the Illumina platform. We did quality control, sequence alignments, variant calling, and annotation of the gene. So this table is showing the characteristics of the participants that were recruited. And the highlight here is that, as you can see, study cohorts, both cases and controls were homogeneous. There was no difference in terms of age, sex, and prior vaccination. Now, after analyzing the full-length gene in the cohort, we identified 30 unique variants. These are single nucleotide polymorphisms at various regions of the gene. And all the variants were in Hardy-Weinberg equilibrium. And then we used a logistic regression to assess whether these variants were associated in any way to HBV infection, and found that 5 of the variants, which are up here, were associated with increased risk of HBV infection, and the 2 down here were associated with protection. And these are the RS numbers, which are unique names given to the genes. So each RS number represents a polymorphism at a particular location. And this figure shows the distribution of the various genotypes in the 7 variants that we found were associated with HBV. And these ones are those that are protected. These are the protective variants, and the rest were the risk variants. Subsequently, we wanted to find out whether the variants, the 7 variants, had anything to do with liver health. So we used generalized estimation equations to assess the relationship between the variants and these 4 liver function parameters, ALP, ALT, AST, and LDH, and found that this variant, 575027, was significantly associated with reduced, lower likelihood of having elevated ALP, and the same with ALT and LDH. The variants that were associated were variants that were found to increase the susceptibility to HBV infection, and at the same time, they were the likelihood of having lower levels of ALP, AST, LDH was also established using the GEE, which means that although these variants are associated with risk of HBV infection, they are also protecting the liver from damage. And we all know that hepatitis B is eliminated from the body through immune-mediated hepatocyte damage. And so this means that these variants are somewhat promoting persistent HBV infection. And we wanted to find out if inheriting these variants together compounded the effect. And so we established a haplotype block using the 7 significantly associated variants and found these 2 blocks to be significant. As you can see, the LD scores here are 71 and 72% in percentages. So, and from the block 1, which is RS 617819 and RS 645419, had 2 of the alleles significantly associated with increased risk of HBV infection. So the GG and the GC genotypes were associated with increased risk of infection, meaning that if you inherit GG, if you inherit either the GG or the GC together, the effects of the SNPs are compounded. So the key findings are that we identified 5 risk variants and 2 protective variants, and we realized that the risk variants were associated with reduced a lower likelihood of having elevated ALP, ALT, and LDH. And we also were able to identify 2 genotypes in the haplotype block which were strongly associated with the increased odds of HBV infection. The limitations of this study were that our numbers were quite low, and so it would be good to replicate this study in a larger cohort so that we can further establish the findings, and also to do some functional studies to find out the exact roles of the variants that were identified. Here are a few references, and I want to thank the HEPMAL team, the study participants, the various institutions represented here. Thank you.
Thank you, Dinah. It was a wonderful presentation. I really enjoyed it. Any questions for her? Please, there's one hand up here already. Okay, please go ahead. Thank you.
Okay, thank you very much once again for the very beautifully rendered presentation and interesting study. My question is this. We found some protective variants, right? And some that we think are not. I don't know whether you also try to do some investigation into other characteristics of the patient, socio-demographic, clinical, other details of the patient, to see whether we could actually have idea of the kind of people that may be associated with the protective, and maybe the kind of people, at least empirically, that we could suspect that they may have the other variants. Because this can also help. For example, maybe tribe, you know, and some other characteristics, socio-demographic, you know, clinical, other things like that. Maybe they smoke, whether they drink, those things. It can help us to have more understanding of the impact of these variants. Thank you.
Okay, thank you for the question. So, as I mentioned earlier, this formed part of a larger study, and as part of that study, an extensive demographic data was collected, and but we did not collect ethnicity. What we did collect was nationality, and majority of our cohorts were Ghanaians, but we did not collect ethnicity. However, in terms of their alcohol intake, we did collect that data, and this was data. Questions asked the patients, do you smoke, do you drink? But we did not analyze the genetic data in that context. But it's worth looking at.
Okay, thank you. There's another hand up here.
As a follow-up on what you just said about alcohol intake, did you really assay GGT? Since it's alcohol and whether to assess whether it has any effect on their liver function as well, alcohol and the GGT as well?
Yes. So we the liver function test we did was the full panel. So there was GGT. However, we had previously analyzed liver function between the cases and the controls, and GGT was not a factor. It was not significantly different between the two. That's why we didn't include it in this analysis. Had an effect on the GGT. Okay, we didn't stratify it like that, like just alcohol and GGT. No, we didn't stratify it like that. But we just didn't include it here because when we look at cases and controls, liver function panel, GGC was not something that was significantly different. It was the same in the two groups.
Okay, Dr. Labi's hand is up.
Thank you. I was wondering about the controls. How did you recruit them?
So this study was looking at, the larger study was looking at hepatitis B and malaria co-infection. So a major factor in the control recruitment was location matching. So what we did was, if we recruited an HBV positive person, we asked them to bring somebody from their household who was not positive. When that proved a challenge because of stigma and people did not want to let their family members know that they are tested positive for Hep B, we tried to find people within their location and recruited them as controls. And we made sure that they were not hepatitis B positive and they were clinically assessed to make sure they didn't have any liver condition.
So when you say location, you mean their places of residence?
Yes.
And not the hospital setting?
No, because the malaria aspects needed people that were living in the same locale because of entomological inoculation rates. Because there's a malaria aspect, you don't want your controls to say that we live in an urban area and we have fewer mosquito bites, and your cases to say we live in an area where there are a lot of mosquitoes. So the location matched had to come in there. And this study used the samples from the same study.
Thank you.
Okay, thank you. Any further questions for Dinah? Any more points of discussion? So, Dinah, all these patients were hepatitis B positive or?
Cases.
Cases, yes. So, maybe I was a little bit confused. They still had protective variants? They had variants that meant protection or, yeah?
So, yes. What this means is that the protective variants were more represented in the controls than in the cases. So majority of the protective variants were found in the controls.
Yes, okay.
There were a few that were in the cases, but that's where the statistics come in. So majority was in the controls.
Yeah, this was my point of confusion. All right, thank you so much. If we don't have any further questions for Dinah, thank you very much for that brilliant presentation. So go to our next one.
Okay, thank you. So take over. So the final speaker of this breakout session is Dr. Desmond Opoku Tiamwa, diversity immune cell profiles of two Ghanaian ethnic groups associated with specific lineages of Mycobacterium tuberculosis complex. Okay, thank you, Desmond.
Good evening. Good evening, ladies and gentlemen. My name is Desmond Opoku Tiamwa, and I'll be presenting on behalf of my co-artists on the topic diversity in immune cell profiles of two Ghanaian ethnic groups associated with specific lineages of Mycobacterium tuberculosis complex. So since its declaration as a public health emergency in the year 1993, tuberculosis continued to pose a considerable health challenge across the world, despite the many control strategies and advancements made. So, and globally, it is caused by the Mycobacterium tuberculosis complex, and due to this, we have it being grouped into. Thank you. So Mycobacterium tuberculosis is caused by is caused by the Mycobacterium tuberculosis complex, which has a population structure that includes 9 human adapted species and 2 adapted species. The 2 adapted species usually it's caused by the Mycobacterium tuberculosis sensu stricto and then Mycobacterium africanum. Phylogenetic analysis of the TB further classifies the Mycobacterium tuberculosis into lineages, which are globally distributed. And this, because of these differences in genetic diversity, so phylogenetic analysis further classify the TB into lineages, and these lineages further influences how the microbacterium is distributed, how severe the disease will occur in an infected host, and then how well an immune response is elicited upon infection. Microbacterium africanum, which comprises of the L5 and L6 lineages, is usually restricted in West Africa, whereby the lineages, whereby the L2 lineages is more susceptible to the peoples in China, and L1 usually affects people around the Indian Oceans, and L3 centered around Central Asia and Western Asia. And as I said earlier, lineages will actually, lineages will, the lineages, so
Everything fine?
Do you want to start? Yes, try and continue. Yeah. Take your time. I'll just cancel the time and start. Take your time.
Okay. Good evening, ladies and gentlemen. My name is Desmond Opokuntiamoa, and I'll be presenting on diversity in immune cell profiles of two Ghanaian ethnic groups associated with specific lineages of Mycobacterium tuberculosis complex. So as I said earlier, after its declaration as a public health emergency, it continues to pose significant health challenge in our health systems. And globally, tuberculosis is caused by the members of the Mycobacterium tuberculosis complex, which is made up of 9 animal adapted species and 2 human adapted species. And the two usually that infect human are Mycobacterium tuberculosis sensoscripto and M. africanum. And a phylogenetic analysis further reclassifies the MTBC into lineages, which are geographically distributed. And these lineages will influence how the MTBC is distributed globally, how severe an infection will be upon infection, and then the type of immune response initiated. So for lineage 2,
Let me present it for you. Okay. So, this is a phylogeny. Okay, the global phylogeny, TB, the lineages that we have for TB, they are geographically distributed. So if you look at this graph, you have the brown and the green concentrating in West Africa. You have the purple in Asia. So we have lineage 2, which is the blue lineage. Is found mostly in the Chinese. If you look at the lineage 1, which is a light purple, you find that it's around the Indian Ocean. So you look at the rim of the Indian Ocean, you find the lineage 1 over there. And the lineage 3 is also found in the central Western Asia. But when you look at what I showed initially, the brown and the green lineage, they are found in West Africa. And interestingly, we don't know what is causing the limitation of these lineages in West Africa. And West Africa has 6 out of the 9 lineages, making it the highest diversity of MTBT strain. So it puts West Africa in a unique position. And studies in Ghana has found that the Macrobacterium africanum, that is the brown and the green lineage, which are also called the lineage 5 and lineage 6, these lineages have been found to be associated with an ethnicity. And these are as a result of 2 independent studies in Ghana. So why is this happening? Why are West Africans associated with lineage 5 and lineage 4 and lineage 6? So the question is the immune system. Let's look at the host pathogen interaction. When someone gets TB, when it goes through the immune system, there are 2 possible outcomes. There is a spontaneous healing when somebody inhales the bacteria. And the other is that it can also lead to acute disease. When it leads to acute disease, sometimes then the third option is there's a containment of the bacteria. The bacteria can be inside the lungs and it will be contained. The immune cells will surround it and contain it. And in this system, it's called the granuloma. So usually when somebody has HIV and then the person has the granuloma, it's surrounded by the CD4 cells and the immune cells, and your CD4 cells go down. You remember the presentation by Jonathan. Then you get the TB. So you see the association between the TB and the HIV. So we want to look at what are the immune factors, the specific immune responses that are playing this relationship, this important role. And the host immune cells, such as the macrophages, the CD4 cells, and the CD8 cells that are contributing to the difference in the pathogenesis. And also what this calls for studies that involve geographical specific areas, because we need to situate it. What we are seeing is peculiar to West Africa. So therefore the research question was, will the proportions of the immune cells of the 2 ethnic groups differ in response to TB? So this is a section of a larger study. Then we hypothesized that based on the sympatric association, when I say sympatric association, I'm talking about the relationship of lineage 5 and lineage 6 to West African hosts. It's looking like we are coexisting with the pathogen in a very nice way. What is causing that? So the aim was to characterize the early phase. When I talk about the early phase, that is the initial exposure of the bacteria to the host, the early phase of TB infection, by looking at the immune cells. So this is what the study was all about. And what we did was to look at cured TB participants and healthy controls. I was very pleased when Jonathan said that he took 100 mils of blood. I wish I could take 100 mils of blood. We could only take 50 mils of blood because of ethical issues. And even with the 50 mils of blood, the group of people that we wanted to take blood from are people who have the TB disease, who have the infection and are in the disease state. But these people are in such a state that taking 50 mils of blood will put them in a very dangerous state. So what we did was that we went into cured TB patients. Even with that, with a large pool of people that we sampled, it's only a few that we could take the blood from, because some of them are still not in good condition. TB is such a bad disease. So we did the outreach at Kolebu Chest Clinic. We called back TB participants who have been cured, and those who volunteered we took. But before then, we checked their HB, their blood pressure, the blood glucose, check pregnancy. If you are pregnant, you are automatically out. We can't take 50 ml of blood. We checked the HIV status. It could be a confinement factor, so we took out the retropositives. So we called about 200 people. About 100 and something responded. We were able to take blood from only 53. So if you be asking questions about the sample size, I've already answered that. So the cured TB cases, we had 33, and then the controls, we had 20. It was very difficult, so please forgive me. Our group have decided that with this presentation, we will classify it as the ethnic group, we will classify it as group 1 and group 2, because whenever we put the ethnic groups there, it masks the presentation. Everybody's attention is on the ethnic groups. So please forgive me. So what we did is that after the blood, we isolated the PBMCs, we cryopreserved them, and when we were ready, we recovered the PBMCs, did antibody staining to isolate CD14 monocytes. Then went ahead to look at to mature them to macrophages. For other parts of the study, we did macrophage infection assay, but for the purpose of this presentation, I want to end here for the presentation, because we want to look at the population of the immune cells. So this is the flow cytometry gating strategy that we use for the lymphocytes, the monocytes in the PBMCs, and the sorted monocytes, and the monocyte-derived macrophages. So this is the demography of our participants. For the controls, we were able to control them quite well. Looked at 10 and control the ages. For the cases, as you know, it was quite difficult for us. We had, for group 1, we had 18. For group 2, we had 15. We couldn't balance the age for the gender very well but it was pretty okay. So these are the distribution of the. And these were the lineages that they were infected with previously, before they were cured. So phenotypically we looked at, and quantitatively we looked at the T cells first, and we found that the group 1 had more CD4 cells compared to group 2. This we're looking at the phenotypically. Then we said that, okay, is it really translating into the count? Then we looked at the count, and again, it showed in the count. And we actually looked at the MFI, the median fluorescent intensity, and it's also confirmed for the CD4 cell count. So meaning that there's a clear distinction when it comes to control for the two ethnic groups, the donors that showed up, there was a clear difference between the CD4 cell population. But when we looked at the cured TB case, we didn't see the same pattern. What could be happened? These have been infected, their immune systems are flared up, it's all over the place, and they are trying to adjust the system. So it was expected to see that we cannot see the same pattern that we are seeing in the controls, the healthy controls that we use, because of the immune system that has responded to the infection to the different lineages. We went ahead to look at the PBMCs. We looked at the CD14 and HLA negative and DR positive ones. We checked throughout, there was no significant difference between the two ethnic groups. And for the cured, as usual, we didn't see any difference between the two ethnic groups. But when you look at the median fluorescence intensity for the PBMCs, where we look at the CD4 positive, HLA negative, DR positive, we again saw that the ethnic group 1 was showing high levels of those cells. And this goes to confirm what we've seen in our studies, that those in ethnic group 1 are mainly associated with the more virulent form of the TB pathogen. Because the less virulent, their immune system is able to beat it down. So it is only the more virulent that is able to infect the group 1. But for group 2, the less virulent one, is able to get across to them. But to understand this further, we need to also look at host genetics, which one member of our study is also looking at. So in conclusion, the difference in CD4 cells, maybe the CD4 count and what the phenotypic and the quantitative may be used as markers to gain some understanding of the TB pathogenesis between the different hosts and the different lineages. However, we need further studies to, as I talked about, the host genetics to understand it. These findings provide inferential information on the role of the immune cells on TB disease outcome. And therefore, we need to further study the kinetics of these immune cells. We need further studies to understand them. And this will help us to characterize the early phase of TB infection very well, so that we understand how we can tackle this bacteria, this enemy, which has been with us for a long time, even in the Bible days. Thank you very much.
Okay, thank you, Stephen, for a nice relay. Now, open for questions and comments. Okay, we have hands from the front. Yeah, okay, please, sir.
Good evening. Hello. Please, my question is with regards to the ethnic groups. So you made mention of two ethnic groups. So does this, in Ghana we have a lot of several ethnic groups. So are you just dealing with two ethnic groups that the mycobacterium is isolated to or strictly related to, or you are just taking two ethnic groups out of the several that you have and doing your studies with those two only?
Okay, thank you very much. So the two ethnic groups is not just out of random. These two ethnic groups are based on our previous list. So one ethnic group, the ethnic group 1, we saw that they are more associated with lineage 4. Then ethnic group 2 is more associated with lineage 5. So based on those two independent studies, then we built our hypothesis based on the findings we had previously. It wasn't random, but based on what we've seen, the association, that is where we went ahead with. People who have been with our, okay, so you maybe through other questions then.
Okay, next question, sir. Yeah. Oh, please.
Thank you very much for a very interesting presentation. Again, being a clinician, my question has to do with these findings. Of course, you haven't told us what the ethnic groups are, but I think that for management's sake, do you think that the conclusions of your study will influence management? Because sometimes we need to go even to do routine screening. And when we have had to go into certain communities, we actually find that there are people there who have TB and they're at home. And anytime a child has TB, we always see that there's an adult somewhere who has TB. Mm-hmm. So do you think that your study will influence management?
Yes. So when we look at TB, our studies that we have reported over the years, and how we compare the ethnic groups to the various lineages, initially we thought that if you are infected with TB, you are infected with TB. Why do you want to worry yourself about the lineage that you are infected with? But when we went ahead to look at the different lineages, and they were telling us different stories about how they infect people, how they behave, and how genetically they are different, and how their disease outcome could be different, and how even lineage 6 is associated with HIV patient. And it goes to look at the fact that if someone, and these lineage 5 and lineage 6 are considered the weaker version, or the weaker, let's say the weaker members of the family. And when the immune system goes down to, the weaker ones can get you, the stronger ones can get you, everybody can hit you. So with the knowledge of this, we are able to influence treatment outcome, or to influence management of cases, and tell them that, hey, if someone with HIV has lineage 4, the person is in more trouble. But if the person is with lineage 6, at least we know that it's less virulent, so we can manage it in some way. Because sometimes when you are giving the TB drugs and the HIV drugs, I know the clinicians, sometimes you manage you need to use different versions of different drugs, especially the rifampicin. You have to use rifampicin and other parts of different derivatives of the drug. So that helps with management. Maybe before I end my talk, I'll tell the ethnic groups.
Okay, I want to learn more about the sampling so that maybe possibly we can implement it some way. In normal blood banking issue, there's a cutoff of sample collection. Mm-hmm. But in your case, did you use any cutoff? Because the samples that you took were beyond the normal diagnostic volumes, 5 to 10 mils, they're about 50. Other person used 100. Was there any cutoff that you used? And how do you sample? Is it multi-sampling, container, or you use different method? And also, in regards to the TB, did you by any chance look at DRTB or they were all MTB?
Thank you very much. So we avoided DRTB so that it will not be a confounding factor. Sorry, I forgot to mention that. So it was something that we avoided so that we wouldn't. Concerning the sampling, what we did with the sampling was that, you see, the ideal sample size that we wanted was 100 mil. Yeah, so, but it was, the ethical boards, it was so difficult for them to grant us the 100 mils. So we wanted 60 mil. We bargained, we went to STC, we bargained, and they gave us 50 mil. So we managed with the 50 mil. The reason why we needed the huge volume was that we want the PBMCs. They form a very minute volume of the blood volume, about 9 to 11% of the total blood that you are extracting. So if you have a huge volume of blood, the PBMCs that you are getting is quite little. And these PBMCs, you need them to get monocytes. And even the monocytes are also a subset of the PBMCs, which are very small. Then the monocytes, we need to mature them or derive them to macrophages. And the more monocytes you have, the more likely you are supposed to get the macrophages, because some of the cells will die along the way. So you need large volumes of PBMCs. As you go down, everything begins to decrease and begins to decrease. Because if you start with a very small volume, by the time you get to macrophages, you have 0 cells. So that is something that we looked at.
I'm pragmatic. Okay, so now, we are a little bit running short.
Yes, so we are done. We are done. We can have further discussion.
It's not a question.
Okay, all right.
For the sake of clarity,
yes.
I didn't evaluate this presentation because the original person who was supposed to present was a lady.
Okay. Was a lady.
Amanda. That's what we have for the evaluation. And then and then you have Desmond coming in. And
all right.
So if I can explain to you what happened. So Amanda has traveled. So Amanda should have presented. Desmond should have presented tomorrow. And just before, just a few minutes during this presentation, they asked him to present.
Exactly.
That's how he was startled.
Exactly.
Yeah, so he's not even on the scoring sheet. He's not even on the program.
Yeah.
To present.
You see, so my problem is when I took a look at the presentation after you took over. You see, the only person who could have presented that was a person who actually prepared the slides. So let's take a moment and absorb Desmond. Don't feel bad about yourself.
Yeah. Okay. Thank you very much.
So he was
He
was not supposed to present. So it's not his fault. Thank you.
Okay.
Yeah. And if I may add, so the ethnic groups are the Akan ethnic group and the Ewe ethnic group.
So which one is
so
you
read our papers and find out more.
Which one is you one and which one is you two? You didn't tell us that.
Please read our papers. You find
out. Oh, please. Thank you very much, Kevin. Thank you. And thank you, Desmond, for the attempt. Thank you.
Okay, so now we have this breakout
session.
So
please, we'll do a short summary and then we have closed. Please, let's calm down. Thank you.
Okay. Right. So we had this breakout session 1, and it was quite cross-disciplinary, I think, because we had many pathogens. We had co-infections, like a pathogen coexistence, cross-species. It's a really, a challenging session to comprehensively understand. But I think that's the beauty of it, and I really feel that these types of sessions it should it's better to increase, I think, instead of really specifying on a given, for example, pathogen or a given slot of an immune response. And I think we shared a really academic experience, and I would have many additional questions to ask each of the presenters. So it was really a privilege to together join you on this session. Okay, thank you for the participants. Thank you.
All right. Thank you all for an exciting session. I mean, I know I can tell that we have all had an exciting time and we have really learned a lot. So thank you all for the discussions and for all the contributions. And so the program, the ARM continues tomorrow, and then Dr. Abana will take us then.
Okay, thank you so much. Chairs, we are so grateful. Thank you so much. Well, like she said, today's program has come to an end, and we continue the ARM tomorrow. We start from 8:30 for registration. Then at 9 a.m., we will start with the AMET Symposium. So please come in your numbers for tomorrow's session. As you file out, let's be like the Japanese, and then take any empty bottles that we have left on the seats. Let's try and clean up as we go out of the room. Thank you so much, and safe trip to your various houses. Thank you.